Good morning, and thank you for joining us all today. My name is Stephanie Bolton, and I help lead viticulture research and outreach for the Lodi growers, along with the Lodi Winegrape Commission's Research Committee. 1 year ago today, I stood up here and told you that I have viruses. This is still true, but since then, I have been tested. It's leaf roll 3. We have a really great lineup of speakers for you this morning. Dr. Kent Dana will teach us about vine mealybugs. Dr. Mark Fuchs from Cornell will talk about viruses. Professor Jerry Peterson flew for 38 hours to get here from South Africa to help us with leaf roll virus management. And Dr. James Stamp will teach us about planting material. You get to not only learn something new, but also listen to some cool accents. Each of these speakers was carefully selected because they really care about helping us, they really know what they're talking about, and they are great at communicating to a grower audience. If at any time during their presentations you have questions, please write those questions down on the index cards that are in the center of your table. We'll collect those index cards just before our question and answer panel later in the program. In addition to learning through presentations from our wonderful speakers, you'll have time to interact with the many businesses and organizations which are important to reducing viruses in California. We have associates in Insectary and Parabug in that back corner. They work with mealybug biocontrol insects. We have 2 pheromone mating disruption companies. Sutera will be passing out again this year, thank you so much, free mealybug traps over there. We have every single commercial virus testing lab in California in that back corner. We have BASF, who has a new ant control product they want to talk to you about. VineView is here, and they do leaf roll virus aerial imaging. And then we have 8 CDFA registered nurseries over here. We have Foundation Plant Services, National Clean Plant Network, the USDA Tree Assistance Program, and the San Joaquin County Ag Commissioner's Office, all represented today. So I think you can tell it's very important to our industry that we get a handle on this problem. So why are we here today? We're all super busy. We all took time out of our demanding schedules to make today happen. It's not quite raining yet, so that's not the excuse. We showed up. Here's why we're here. It's simple. We've got a lot of these. Grapes, vineyards, handsome guys named Aaron. We've got a lot of these. Vine mealybugs. Too many, really. Almost all of us have started to see more and more of these red leaves. And all this is costing us a lot of these. So what we did is we formed some teams to try and figure out what to do in Lodi about mealybugs and viruses. The first team we formed was the Mealybug Biocontrol Research Focus Group. It was important for us to have a mix of growers, PCAs, extension, and scientists. This successful group hosted a Mealybug ID field day in August 2017, and they also created the beneficial insect risk chart. Next, we formed the Lodi Grapevine Virus Research Focus Group, also a team of growers, pest control advisors, scientists, viticulturists, and extension personnel, and nurseries. And we're lucky enough to have Professor Peterson and Dr. Mark Fuchs as our experts willing to give us advice and assistance as we navigate our local virus situation. It helps that many members of our team have a financially vested interest in finding practical virus management solutions, and that they are willing to work towards a greater good for everyone. After all, our Lodi Winegrape Commission motto is, "A rising tide lifts all boats." We meet on the first Wednesday of every month at the Velvet Grill Diner in Lodi to discuss viruses for hours over pancakes and coffee. This photo is from yesterday's meeting. We started learning everything we could about virus management from anyone who would talk to us. We were in shock for a while at how bad the situation was. Then we accepted the reality that in California we have prolific, widespread populations of vine mealybugs, and that there is a significant amount of virus inoculum or source points in our vineyards. We also realized that no one locally had a good working virus management protocol. We became determined to find solutions to help ourselves and our fellow growers. It became clear early on what needed to happen. So we wrote a grant proposal for an integrated outreach project focused on grapevine viruses. Luckily, we were funded by the American Vineyard Foundation and the CDFA PD GWIS board. If you are a reviewer or a board member, thank you. This project includes virus testing and the collection of case studies, the writing of virus management booklets as information becomes available, monthly meetings for growers and for the focus group, the formation of demonstration vineyards, and lots of outreach. We've also been successful in bridging the gap between growers and different sectors of the industry as we work together to find virus management solutions. We believe that when we all learn together, we can better work together and we can find solutions faster. Consider yourself involved. Our main strategy right now is to decrease vector populations and to lower virus inoculum. In other words, we need to lower the spread and numbers of vine mealybugs and rogue virus-infected vines out of our vineyards as it is financially feasible. It's going to take a lot of teamwork and help from the growers, nurseries, FPS, the CDFA, academia, virus testing labs, wineries, extension, IPM companies, pest control advisors, and viticulturists, county ag commissioners, and regional associations. Basically, if you're in the room today, we need you. We need to stop pointing fingers and realize that we all contribute to the problem and to the solution, and then decide what action each of us can take so that we can all move forward together. Step 1 is education. Last night, we hosted an intimate grapevine virus influencer dinner where about 20 people talked through their long-term strategy ideas for grapevine virus management in California. Later today, we'll host meetings for nurseries, virus testing labs, and extension, each with growers as part of the conversation. We're definitely making progress in the area of communication between industry sectors. Many of you have been joining us for our virus outreach events. Be sure to give your email address to Julie or Shirley at the sign-in desk if you want to join our new Grapevine Virus email listserv so that you can stay informed. Our virus focus group plans today's indoor meeting for the spring each year, and we also plan a hands-on meeting in the vines each fall so that you can learn how to scout for virus symptoms in red varieties. Last fall, we also learned about mealybug biocontrol, virus testing, and the three-cornered alfalfa hopper. We're realizing that we need to focus our management efforts on these guys: leaf roll 3 virus, vine mealybugs, and ants. Does anyone recognize this photo taken by Mario Salinas, who's in the house today, by the way? It was on the cover of the recent PD GWIS newsletter, and it will hopefully convince more California growers and pest control manufacturers of the need to control ants in vineyards. This little ant is carrying a mouthful of mealybug eggs along a drip irrigation line, off to start a new colony of mealybugs on a grapevine. We've been collecting case studies to investigate the potential role of leaf roll virus in a mystery vine collapse where large patches of vineyards are dying all over Lodi and in other parts of California. Vines push out fruit and appear healthy, and then all of a sudden, in a matter of weeks, the entire vine is dead, dried out, and even splitting along the trunk in some cases. At this point, it is looking like a combination of freedom rootstock and maybe others, leaf roll 3 virus, and a third stress, which could be trunk pathogens, vitiviruses, crop or water stress, etc. Over time, these patches become larger until the vineyard is no longer profitable and needs to be removed. We're creating a grapevine virus workbook for growers, starting with small draft booklets that can be handed out before the final version is ready. This way, we can get some tips to our community now, and we also get a chance for growers and others to help us improve the content. We're sharing hard copies of these booklets with anyone who wants one, and the goal is for other wine growing regions in California to customize them for their growers. The first booklet we made is called Nursery Ordering 101 Viruses, and it teaches growers how to order CDFA certified material, which is actually quite confusing, and then how they can take some responsibility for starting with cleaner material, which includes asking for it, extra testing in the beginning, and good record keeping. If you're planning on ordering plant material soon, you should definitely pick up a copy back there. Our second booklet is on vine mealybugs and is available for the first time today, hot off the press. By taking a draft copy, you agree to provide comments and input. We have 50 copies here with us today, and they come with a new custom vine mealybug flagging tape. Thank you so much for all you're doing to lower the amount and spread of vine mealybugs, to decrease virus inoculum in the vineyards you manage, and to educate yourself. We are grateful to the CDFA's PD GWIS Board and the American Vineyard Foundation for funding, and UC Cooperative Extension for sponsoring our room today, and to the members of our Lodi Mealybug and Virus Research Focus Groups, steered by the Lodi Winegrape Commission's Research Committee, for all of their dedication to helping their fellow California growers.
Working together in the wine grape industry has been really exciting to see over the last 5 to 7 years, watching wineries and vineyard management agencies that are all competing in the same space, all really group together to solve this problem. I think that's been the most exciting thing to see this morning.
Next up, we have Dr. Kent Dana, the smartest mealybug guy we know. If you own a Lodi vineyard or if your paycheck depends upon Lodi grapes in some way, you should definitely tell Kent thank you today. He released populations of anagyrus wasps in Lodi years ago, which have played a really significant role in parasitizing vine mealybugs here. Let's all give a warm welcome to Kent.
I'm going to give an overview of vine mealybug as a vector of leaf roll. We're not going to talk about red blotch here today, the other red leaf disease, and the different types of control programs we've got. This is going to be more of a general presentation, but I'm going to stick around for the work groups afterwards so we can get down into the weeds about the nitty-gritty details of all of these different control tactics. And I want to thank all the co-authors, friends that I've worked with, colleagues in the lab, and leaders of other laboratories that have helped with this project. So vine mealybug is one of a number of invasive species. It's one of 4 invasive mealybugs. We've got the vine mealybug, the obscure mealybug, the long-tail mealybug. Even the Gills mealybug is not from California. The only California area mealybug is the grape mealybug. It was the #1 vector of leaf roll before the vine mealybug arrived. Vine mealybug is much different than these other mealybugs in terms of its effectiveness at moving the leaf roll around, particularly because it's got so many generations per year. And the crawler, the smallest stage, is the best stage in terms of efficiency in moving it. Since it's got probably 6 to 7 generations per year here in Lodi, you've got a lot more crawlers, a lot more chance for the vine mealybug to move the pathogen around. So basic outline, we'll look at mealybugs as invasive species, their damage, not just direct, but how they work in terms of being a vector of leaf roll, biological controls, mating disruption, chemical controls. Then I'll try to bring this all together to talk about practical applications and conclusions. So as I mentioned, vine mealybug is one of these invasive pests. The next one is going to be the spotted lanternfly, which is now on the East Coast, can be devastating of grapes. Hopefully the Rockies, the mountains, the deserts keep it from getting over here. Vine mealybug, like the obscure mealybug, came from someplace else. This is the obscure mealybug, 3 generations per year. We find it in Lodi, although its numbers now are harder to find because of all the applications for vine mealybug. It came from South America. It spread throughout the world, mostly on nursery plants. When you go into any of the shopping malls and you see things like ficus or on the umbrella leafy plants, and you see mealybugs on it, it's commonly the long-tail mealybug or the obscure mealybug. So, spread throughout North America. Interestingly, in South America, they don't have as many problems with the Argentine ant as we do up here. That's because the Argentine ant in South America keeps its own numbers down fairly low. One colony of Argentine ant recognizes another colony just down the street as an enemy, and they kill each other. Here, we've got one large sister population of Argentine ants that all work together. That's why it became such a pest here. Vine mealybug, in particular, came from the Middle East, Mediterranean region. It's now a pest in South Africa, it's a pest in Chile, it's a pest in Argentina, it's a pest in Peru. If you go to France, they don't consider this to be as much of a problem as we do here. That's because it's probably made up of a bunch of sister species. And in fact, when we got into the weeds on this, looking at the molecular work, we can see the California and Mexican population. This is kind of a phylogeny tree looking at the relationships of different populations of vine mealybug. California and Mexico have got a very, very similar population. So you would assume it came from the same spot. When we started working with this, we thought it came up from Mexico. I went to Mexico to work with colleagues there. They thought it came down from California. Well, in fact, we know which farmer brought it in. He farms in Coachella. He farms in Hermosillo, the two spots it started. We know the first find of vine mealybug was in his next door neighbor's spot, and he had bragged that he had gone to Israel and picked up some wood with table grape material that was better suited to the desert region. And in fact, when we look at material from Egypt and Israel, we can see it was very, very similar to a population from Israel. And we see how far apart this group of Israeli-Egyptian vine mealybug is from populations found in Europe. So that's one reason why it might be a little bit harder to control here. It might be a little bit better suited to hot climate. We can see for Gerhardt, the South African population probably came from Europe, probably from Spain or Portugal. Argentinian population probably came from Spain. And we've got two other groups here from Turkey and Tunisia. So each of these different populations will have different biological characteristics, making it easier or harder to control depending on how many generations they have, how much fecundity they have, meaning how many eggs they've got. Once in California, we found it in Coachella. Walt Bentley tells the story of looking at those 20, 30 vines that were infested, discussing whether or not they should pull the entire vineyard out or just nuke it. The decision was, let's just put on a lot of lanate and lorsban and it will probably kill it all. It then made it into Southern California, around Bakersfield, around Fresno area. We now suspect these were infested table grape clusters that could not be marketed, so they were sent into the Central Valley to be crushed for juice grapes. We now know that the vine mealybug, a small percentage, can actually survive the crush going through wine grapes, and people would take that crush and spread that pumice out on the dirt roads to lower dust. So a tiny percentage can survive, so we probably got, because the first finds were around juice-producing plants, we probably brought it up from Coachella, spread it on the roads, and it made it into the vineyards. And then, of course, there was explosion later in the early 2000s because it did make it into the nursery material. The nurseries were doing what they were supposed to do to control for exotic pests, because everyone at this time was concerned about Pierce's disease and glassy wing sharpshooter. And the control for that was to spray the nursery material with Pravado, imidacloprid as a foliar form. That's great for glassy wing sharpshooter. We know that doesn't work for vine mealybug. You've got to use Admire or one of the other systemic neonicotinoids, or Belay, a foliar neonic. So what happened was that it got into the nursery material, it got down on the roots, and we started planting vine mealybug throughout California. I mentioned it's a lot worse than the other invasive species. I take this because of 4 different reasons. First, it causes more damage just in its feeding because it's got more generations per year. Grape mealybug, about 2 generations per year. Obscure mealybug, about 3 generations per year. Vine mealybug in this region, about 6 to 7. It can have as many as 8 generations per year down in Bakersfield. It's going through a generation right now. Grape mealybug has a diapause. It stops sometime in September, October developing, just stays as an ovisac or a first instar. Vine mealybug in January, if you kind of get a nice warm day, it'll start to feed, it'll start to develop. We oftentimes don't see this because we think that nothing is happening underneath the bark. But we're going through a generation underneath the bark in January and February, and so we get these off-sync populations. Right now, I can go to any of your infested vineyards, and I'm sure I can find crawlers, second instars, third instars, and even some adults. And that makes some of our chemicals less efficient, especially for the organic growers, because those materials really target the first instars. It also feeds on the grape leaves far more prevalently than the grape mealybug. That allows it to get up above the cluster. It allows it to rain honeydew down on the cluster, causing more damage. Much more important for table grape growers than when we consider it as a pest, just because of its ability to vector. So where we've got typically, the grape mealybug would have populations just like this. The vine mealybug can really explode in densities. But what we're interested in is its issue in terms of being a vector. Classic picture from Deborah Galino, close-up of a leaf infected. Here's some of the work I did with Rodrigo Almeida's lab. First bit of information you have to remember as any of these mealybugs, as being vectors of leaf roll, is they can acquire the virus very quickly. So within 1 hour or less, they can pick up the pathogen. It goes up to about 24 hours. That's what that graph showed, and it kind of maxes out. They can inoculate the pathogen very quickly. Within about an hour, it can move it, once feeding on the vine, into a clean vine. So an hour acquisition, an hour inoculation. That means for me that if you're using one of these great materials, any of the systemic materials, Movento is a systemic material. The neonics are typically systemic materials. The mealybug has to feed on the vine in order to die. With Movento, it has to feed for quite some time to die. So we always say wait 3 weeks, wait 4 weeks sometimes, depending on where that mealybug is. So remember, acquisition and inoculation within 1 hour, and we've moved away from the lanates. We've moved away from the toxic contact materials to systemic materials. So you can have mealybugs brought in by birds, by equipment, by air. The crawlers you're not going to find anyway because they're so small. They can feed, they can transmit the pathogen, and maybe 3 or 4 days later they die, but as a vector it's done its job. One of the other things I like to point out is that so many vector-pathogen relationships, when we've got an insect and a pathogen, it's a long evolved situation. Not so with these mealybugs with leaf roll. This chart just shows the relationship on the left of different leaf roll viruses, and when this was done, they were going up to 8 or 9 or something like that. And on the right, these are just species names of different mealybugs, so I'll give some pictures of the 4 we're talking about. And we see all the arrows going basically to leaf roll 3, because that's the most common one, so it's the most tested one. But the bottom line is that there's no real evolved relationship. Almost any of these mealybugs can be a vector. So right now, if you're in El Dorado County, you're worried about the Gills mealybug. This was first found in California on pistachios, then almonds. Now it's on grapes in the foothills. We tested it. Yes, it also is a vector of leaf roll. So there's no evolved relationship. Any mealybug should be considered bad. Another point that I like to pull out, we tested these different species, and we can see here in the laboratory they're between 5 and 50% efficient. In the field, between about 1 and 30% efficient. Well, what that means is these are very effective vectors. Glassy-winged sharpshooter has to feed and probe and feed again and probe again before it transmits Pierce's disease. We're taking one crawler, something you can barely see, and putting it on a vine, and sometimes as much as 50% of the time, that one crawler is transmitting that pathogen. So it's a very efficient vector, which makes control a little bit more difficult, because it brings the number of acceptable mealybugs down to such a low level, I don't think we can attain that with insecticides, biocontrol, maiden disruption. I think we have to focus on roguing, because we're never going to get the mealybugs down to a level that's acceptable for them to control as vectors. There are some differences in efficiency between mealybug species. We're looking here at young mealybugs, old mealybugs, and we're looking at, on the top, vine mealybug versus the other invasive species, obscure mealybug. And if you look at the inoculation, the graph on the right, it just shows that the vine mealybug was a more effective vector than the obscure mealybug. I've been trying to figure out why that might be, why the crawlers might be more efficient than the later stages. I think maybe it's because the vine mealybug, because it has so many generations per year, it feeds more. If you go from a baby to 6'5" in 4 years, you're feeding a lot more than if you're doing that in 30 years on a day-to-day basis. So maybe by feeding more, they're acquiring more of the pathogen. I think the crawlers might be more efficient because they might just be sloppier. They've got this thing in their esophagus, this pump to bring the fluid in, goes to the digestive system and comes out as honeydew. And maybe as an adult, that pump's a little bit more muscular, a little bit better at bringing the material up and not being sloppy, like a kid with a straw blowing bubbles back into the milk, something like that. These details we haven't worked out, not that important. What is more important is that they do lose the pathogen with every single molt. So if it feeds on an infected vine, it's not going to pass that pathogen on. If it goes to a clean vine, molts, it's not going to pass it on to its offspring. Some vectors can do this. It makes control much, much harder. So once it's fed on an infected vine and acquires the pathogen, 3 or 4 days later, it loses that virus within itself. One of the other aspects of this, and I'm going to run out of time, so I'm going to start speeding this up. One of the other aspects that we always get questions on is how soon do we see symptoms after the mealybugs feed? This becomes very important for new plantings. Did I get it from the nursery? Do I have mealybugs in the ground that are feeding on the roots, coming up, and now I've got the pathogen? So what Kai did with Rodrigo and myself is that she went out and we inoculated 10 mealybug crawlers per plant on July 19th. And we can see the arrow following that, the little circles at the very bottom of the graph. We saw no symptoms that year. So a July inoculation, no disease symptoms the rest of that year. The next year, we were starting to see symptoms around June, and the following 3 years, around mid-May, we started to see symptoms. So if you are inoculated this year, in the middle of the summer, you're probably not going to see symptoms until next year, and then you'll start seeing them around May or June, every single year thereafter. So, what about control? I'm not going to go into detail on any of these subjects except for chemical control. We'll start off with natural enemies. There are a number of good predators out there. Many of these you can purchase. We've got associate insectaries here. Brett's an expert of all of these. He can tell you the pros and cons. So I'll just give you a generalization. We've got the going around the clock. We've got the green lacewing. There's also the brown lacewing. You can't purchase that. It's typically more common in the spring than in the summer than the green lacewing. We've got the mealybug destroyer. It's got the larva that's got the dreadlocks on it where it looks like a mealybug, so it works a little bit better in the presence of ants. Years before, I've shown videos of all these natural enemies, so we're just going to talk about their basic biologies here. The mealybug destroyer works better in the presence of ants, but it tends to leave if the mealybug densities get a little bit low. There's other kinds of predaceous ladybird beetles that are native to California that also have got larvae that have the kind of dreadlock look to them as well. Sometimes we release the mealybug destroyer, we see these others. All of their issue is overwintering and the density of the mealybug. The little orange larva is a sesodemyid midge. These are common in vineyards with large numbers. And in fact, that's what I think is one of the issues with most of these predaceous natural enemies of mealybugs, is that to stay in the system, they like to have a vineyard with a lot of mealybugs in it, because that's where they're searching to put their eggs. Remember, these tiny, tiny little first instar mealybug destroyers can only crawl around on a single vine. They cannot go 20 vines down or across the road or across 5 rows. They just aren't that big. They're not that good of dispersers. So the adult female, mom, will fly away and try to find a vine with a lot of mealybugs on it. They're not lost to the system in Lodi, but they might end up in your neighbor's vineyard if he or she has more mealybugs. That's why area-wide controls are oftentimes important. I really like, at low densities, the parasites a little bit better. This is Anagyrus pseudococci. It was first imported in the 1920s, the 1940s for the citrus mealybug, the Comstock mealybug. We brought material in from Spain, which I like a little bit better. It probably is a different species. We're not separating these out yet, but it attacks the smaller stages of the mealybug. That's why I think it's a little bit better at controlling mealybugs throughout the year. There's also this Coccidoxinoides peregrinus. We brought this in with von Walton from Stellenbosch. It has a bigger role, I think, in South Africa than it does here. We're finding it here, it's established here, but we don't see it in the kind of numbers as we see Anagyrus, in part because right before it gets to a pupa, the mealybug kind of senses something's going wrong, and it drops from the vine or it goes in the cracks and crevices on the bark, and we just don't see the pupa on the leaves. These parasites do have problems. All these control tools will never get you down to zero. We can't eradicate mealybugs, and I'm going to stress that again. So, for example, Anagyrus is the best of the parasitoids that I think we've got. One of its issues is that it starts too low in the season. We see here in May, parasitism is less than 10%. That's because it's got this overwintering biology where it doesn't want to come out from the mealybug as an adult until the mealybugs are out on the vine. It doesn't search well for mealybugs underneath the bark. So it just evolved not to be present, not to be searching until the mealybugs are starting to go out and onto the leaves. That's good for it, that's bad for us. It reaches high levels of parasitism late in the season, but remember, as a vector, as an efficient vector, we're really going for zero. So it helps, but it's never going to drive you down to zero. There's always going to be mealybugs located underneath the bark that this natural enemy doesn't get to. And so ants are also a problem for almost all of these predators, especially the parasitoids. We've shown data before, if you've got ants, you've got less parasitism. What I'm showing here are two types of controls. We worked with the liquid baits in tanks. Typically, you can purchase these things. They've got commercial products. We should have gotten a Section 18 for boric acid. I still want to do that. Eventually, I will get to it. But it became labor intensive. We've also got these gels that are being tested. These also have to be broadcast spread. This also became labor intensive. So all of these ant controls cost a little bit more money than they work, but they cost a little bit more, they take a little bit more time. So that's adding to your mealybug controls. We worked with Jocelyn Miller, a really great pheromone chemist out of UC Riverside. He developed pheromones and synthesized pheromones, identified and synthesized all the pheromones for all the mealybugs in California. And the vine mealybug was so cheap compared to the great mealybug to produce. Initially, we were just trying to do this as a monitoring tool. And we did this before vine mealybug was even up here in Lodi. And Walt Bentley and I would go around and give talks saying, hey, use these traps. These are amazing. Use these traps because the mealybug's going to be here one day. And when you find it, tell everybody, and we can go in and we can spray it. Well, you guys were not so vocal when you found it. So it got up here, again, moved on the nursery, and all of a sudden it was in Napa, Sonoma, and eradication was just not even an issue. Napa tried, and they couldn't control it. So we found that not only was this good for monitoring, but it was so cheap to produce. The base of this formula is the same base in the perfume Charlie. And so what happened was that there were groups, a company in Japan that was mass producing these perfumes, and it was a simple one more step to mass produce this pheromone, that synthesize the pheromone. Still, it's expensive, and we'll talk about that later. We initially tested this with Sutera products. Initially, it was a sprayable formulation. We were almost always getting a reduction by at least 50%. The lower the mealybug, the better it would work. We almost always had a trap reduction, meaning we shut down the pheromone traps, which was good, and we typically got a reduction in crop damage, which is good. And this particular trial went from about 9% to about 3%, so about a 60% reduction. We then tested this as a pheromone in a lure, because Suterra initially was going to market this to the organic growers. The encapsulated formulation at that time, they're working on getting it registered for organic growers, but at that time it was not going to be registered for organic growers, so they came up with the dispensers. I'll show you data next with Pacific Biocontrol. It's great to have another player in the market because maybe price competition will lower the price for this a little bit. So Suterra product on the left, Pacific Biocontrol on the right. I'll use same kind of data for both groups. I'll use last year's data from Pacific Biocontrol. We can see here excellent trap shutdown. What I really liked about their isomate dispenser is it clearly lasted the entire season, so we're putting it out next week to put it out really early this year and see if we're still getting trap shutdown in October, November. Here's what I want to point out to get us back on the idea of area-wide control. This is the trial of Pacific Biocontrol. Blue is no mealybugs, red is a lot of mealybugs. We've got yellow was the control, red was the pheromone. The chart down below was wind direction coming from the northeast. This is before we put pheromones out. This is after we put pheromones out. You can see the flip-flop. But the most important thing to see here is that on the windward upside, in the control, a lot of mealybugs, we're getting control even in our control plots because the wind is blowing the pheromone. But on the north side, we're still not getting as good of control because the wind is blowing the pheromone away. If you've got 2 acres, if you've got 5 acres, and you think, oh, in that 2 acres, I'm going to put out mating disruption to knock this back, it's not going to work in those 2 acres. The larger area you use with mating disruption, the more effective it's going to be. So I'd like to see area-wide control using mating disruption and really target the insecticides where you've got bad populations to try to bring that down. So I'm going to finish this off with insecticides. We initially were working on something other than Lorsban because we lost Lorsban. It was actually an in-season product at one point when I started. Then we lost it as an in-season. Walt Bentley got a Section 18 as a delayed dormant product. So then we were looking at something that would be as good as Lanate. We've now lost Lanate. And we came up with Applaud. We've now lost Applaud. And we came up with the neonicotinoids through the drip, which we still have and I think still work very well. Later on, along came Movento. Belay is another good product. There's others, but we really liked this combination of a neonicotinoid and the Movento Spirotetramat because we like to go one product than the other. If you're going to work on resistance management, work with your neighbor because you've got their mealybugs and vice versa. We had to find out how Movento worked. Remember that it's sprayed. Let's see if this will start to go. It's sprayed as an AI. It goes into the leaf and it gets metabolized to Spirotetramat enol. It's the enol that kills the mealybug, not the AI. So the first thing it has to do is get changed in order to work. So it goes through this different string of chemicals, of metabolites. The enol, the one that kills the mealybug, is the first one in the chain. Once it goes beyond that, you've lost its ability. So you want it to convert immediately to the enol and then stay as the enol as long as possible. We use this machine called an HPLC to look at the amounts of Spirotetramat and enol in vines throughout California to figure out why some growers were saying Movento did not work this year. Because again, it's the best product that I can think of. Here's one example. This is 5 days after treatment. These are 20 different vines. In red will always be the enol. In white bar will always be the Spirotetramat. This just shows in this particular vineyard we got 100% conversion to the enol. That's good. That's what we expect. We also see that there was about a 10-fold difference in the amount of the enol in leaves from different vines. So different vines are going to have different rates depending on how they acquire the material, how the spray goes out. Maybe it was our fault where we collected the leaves. Maybe some of the leaves inside didn't have as much of the product as those outside. 72 days later, we still see this difference with some vines having more than others, but we also see a lot of the product still in the vines. So what are some of the quirks with using Spirotetramat, using Movento? In this particular vineyard, 5 hours after application, we've got mostly Spirotetramat. So we know it does take some time to do the conversion. 4 days later, we've got 50% spirotetramat and 50% enol. For some reason in this vineyard, there wasn't 100% conversion. The spirotetramat won't kill the millibug, so half your product was lost in terms of its effectiveness. Maybe it has to do with the stoma being open or closed. Maybe it had to do with the surfactant that the grower was using. Maybe it had to do with vine physiology. These are all questions we're trying to work out. One of the other things to note, 184 days after application, that spirotetramat really still had not converted over. So I think the conversion is immediately when it goes from the leaf into the leaf, rather than going from the trunk back up to the leaf later. I also want to point out that 1 year later, we're still finding spirotetramat enol in the vine. It's at a much lower level, but it's in the vine for about a year. That can be good. It's in there for a long time killing. That can be bad. You get down to a really low dose, and what does that lead to? It leads to resistant possibilities. What about movement from the leaves? These were all data from the leaves. Everyone wants to know, what about the trunk? That's where it's hard to kill the mealybugs. It's really hard to kill mealybugs on the trunk. I haven't found a good way to do it yet. We spent a year working with Movento in different ways of applying it. But when we do find it on the trunk, when we do find it on the roots, it's not that low in parts per billion or parts per million. What we find instead is that it's spotty. So when we spray it on a vine, we collect the leaves, we can spray it on one half of the vine, and 10 days later we find it on leaves on the other half of the vine. We spray it in shady leaves, it's on sunny leaves later. So it spreads evenly throughout the leaf. We spray it on the leaves and we take trunk samples, and half of the samples have 0 spirotetramat enol and half have a lot. So it's spotty, and it's even spottier going down to the roots. We don't know why yet. I'm asked all the time about dose, about rate. We want 8 ounces, 12 ounces, 24 ounces. So we're going way above label rate, and we're looking at 1, 7, 30 days later, spirotetramat on the left, enol on the right. Take-home message is the spirotetramat, the active ingredient, is the way it should be. It goes up with increasing doses. The enol, again, doesn't always follow that pattern. And it has to do, again, probably with vine physiology, something that's going on in that conversion that we don't quite understand yet. I'm asked about post-harvest applications. I prefer to see a plot or something else go on if your harvest is really late. The leaves have to be actively photosynthesizing for that conversion to happen. So in this one, we sprayed really late in a ratty old Thompson seedless, and we have nothing but spirotetramat. We have no conversion. If you're a table grape grower in Bakersfield and you're putting this on in July after harvest, yes, you probably get conversion and it probably has a post-harvest impact. But if you're harvesting in October, late October, I would try a different product. So finally with Movento, we had a number of failed vineyards where they put on Movento. We had terrible populations of mealybugs. We went out with our machine, we looked for enol and spirotetramat. That's where the spirotetramat should be. That's where the enol should be. So far we've done this 4 times. In all 4 cases, there was an application error. So I think the application is really key with Movento. We don't know all the way it should be done, but just as kind of general rules using Movento, if you've got underneath the bark adults that are putting in Ovisac, Movento won't kill those because they're not feeding. They're putting out an Ovisac. If you've got eggs, they're not feeding. It won't kill those. It's not getting down to the trunk in the same dose that it's getting to the leaves. So those mealybugs on the trunk aren't going to die a week after you spray or 3 weeks after you spray. It's going to take a much, much longer to do that. There's differences in vine physiology where this product goes. So in conclusion, lots of different mealybug species in California. South Africa has the same issues. We're unique in that we've got almost everyone's mealybugs. Lots of chemical controls. I listed some of my favorites, but Movento, Belay, Admire, all the neonicotinoids, Applaud, which people are not using as much anymore, Savanto, all have knockdowns. I like probably Movento as the best, but the others work very well as well. I think Movento, Belay is what I'd like to see back and forth every single year. There are excellent biocontrols for grape mealybug, not so much for the others. Talk to Brett, talk to us later on in the breakout sessions on how to use these natural enemies. I like mating disruption, but it's got to be used as an area-wide program or larger acreage. These are all tools. Nothing eradicates the mealybug. So no single control works. Again, area-wide programs are best. Your neighbor's mealybugs are your mealybugs. Their viruses are your viruses. Thank you very much.
Lodi Wine Growers has been great. They do a tremendous amount for this community. Their communication is fantastic with growers in all aspects, and especially most recently they've done an excellent job of giving growers a resource to find information, especially about topics like this.
Thank you. So next up we have Mark Fuchs. He's from Cornell University. I met Mark down at the Sustainable Ag Expo in San Luis Obispo. I think it was 2017. And he gave 3 presentations at that Ag Expo. They were all fantastic, and he really simplified this whole complicated grapevine virus knowledge into really easy to understand concepts. And then he was so patient as I grilled him with questions during lunch, and he's been so supportive of our virus efforts in Lodi, really answering questions for us, and he came all the way from New York to be with us today. So please give a warm welcome to Professor Mark Fuchs.
Good morning, everyone. Thank you so much, Stephanie, for this wonderful introduction. My heartfelt gratitude is expressed to you and to every one of you sitting in the room today for taking time off your busy schedules and participating. And kudos to Stephanie and all of you for the wonderful program that you have launched to collectively try to mitigate the impact of these viruses. So, talking about my presentation, Stephanie tasks me today with the idea of communicating on what viruses are and to provide general information on their properties or some of their unique properties, because when you compare viruses to Pierce's disease or Xylella, the causal agent of Pierce's disease, other diseases that you might be more familiar with, like downy mildew, botrytis, rot, and so forth, viruses have very unique properties. So we're going to walk through these properties and explain you, based on these properties, how can one devise optimal management strategies. And these strategies, as you will see, are very, very simple. Actually, they are so simple that very often we neglect to consider them in a vineyard management program. So, this being said, let's go and start with viruses. What are viruses? Viruses are very small infectious agents. Their size is very, very, very tiny. They have different shapes. They have the genetic information that is quite unique, is very specific to a virus, meaning it can be either RNA or DNA based, and that's a different chemistry, but it can be very unique. And more importantly, not only are they tiny, they cause huge damage, but they're obligate parasites, meaning unlike bacteria and fungi, they can only live and survive on a living organism. The vine has to live in order for these infectious agents to thrive and multiply. So viruses are different shapes and sizes. This icosahedral shape here to the left is depicting grapevine fan leaf virus. The one to the right is depicting, which is a very filamentous shape, leaf roll. That's a particle of leaf roll virus. And in the middle here is a hypothetical representation of what red blotch should look like. And when I mention hypothetical, because nobody has seen red blotch particles in an electron microscope. So when you look at the size, I put scale bars here. They are very, very tiny, meaning that, for example, fan leaf has a diameter of about 10 nanometers, meaning that, to put everything in perspective, I'm about 6 foot tall, all right? If fan leaves were my size, my shoes would be in this room, and my head would be on the other side of the country on the East Coast, just to give you a perspective of how tiny these viruses are. And as I mentioned, they are very diverse in how they carry their genetic information. In other words, what is very key to them being able to survive after they infect a vine. Left again is fan leaf, and you see this icosahedral shape, so you can imagine roughly a basketball, and the ball inside contains the genetic information. For leaf roll, that is shown to your right-hand side, you have the genetic information that is twisted somehow along that spaghetti-like shape. And just in the middle, for the fun of it, I depicted another virus. This one does not go to grapes. It's a virus that infects bacteria, so a bacteriophage, and it's like, you know, coming from a Steven Spielberg movie, right? It looks like an alien, which in this head has the genetic information. So how do viruses infect a vine? And I'm going to take you down really at the cell level, at the individual cell level. As I mentioned, viruses are obligate parasites. So they go inside a cell. They do not stay on the surface of a leaf, of a shoot, or a root. They are inside. So they penetrate, and we're going to go back later on as to how they penetrate the cell. They have to have a hole in order to enter. Because viruses, and like fungi or bacteria, are so small that they cannot and do not encode all the proteins and enzymes that are needed to degrade the cell wall of a vine cell. They need to have a puncture. And very often, in relation to Ken's presentation, the puncture is made by an insect vector, when the insect vector is feeding. So the virus enters the cell, and as soon as it enters the cell, this icosahedral shape of fan leaf will just open up. You can imagine the basketball losing air and will open up, so that the genetic information gets released in the infected cell. And once this genetic information is released, immediately there is a very intimate relationship that goes on between the virus and the plant nutrients, organisms, proteins, enzymes, because this is how the virus survives. It basically hijacks some of the primary functions that are needed for the plant to thrive to its own benefit, because it is so tiny that it cannot survive on its own, unlike a bacteria and like a fungi. The virus has to hijack a number of proteins in order to survive and multiply. So that's what is happening. The genetic information goes there. It is used to make the viral proteins, and then the whole thing gets together again in a very intimate way between plant constituents and virus constituents, and they form this type of factory, if you want, close to the nucleus of a vine cell where the virus propagates. Basically self-clones itself by using, again, constituents of the plant. And after that, you have a multitude of little viruses or genetic information of the virus that penetrated that cell that will form, again, virus particles, and these new virus particles are ready to be picked up by an insect vector that feeds on these cells and to start a new infection of a healthy vine. So that's basically a cartoon representation of what's happening. You have penetration, you have production of proteins, you have replication of the virus, and the virus is then picked up by an insect vector and moves on. But obviously, in order to infect the whole vine, the virus has to move from cell to cell, and from the cell of the organ where the infection started, and invade systematically the whole plant. How does the virus do that? Because again, there the virus is facing a conundrum, because plant cells are adjacent to each other. They communicate, but the communication gaps are very, very narrow to just let nutrients flow from one cell to the other, and very tiny molecules that are even smaller than viruses. So how do viruses move from cell to cell? Well, they're basically tricking the plant. And how do they trick the plant to move from cell to cell? They produce themselves proteins by hijacking, again, some of the functions of the plant in order to widen the very narrow gap that exists between two adjacent cells. So the virus will produce these proteins that will stuck into these narrow gaps and widen the gap so that the virus particles can move through and go from an initially infected cell to a healthy neighboring cell. And once they do that repetitively over time, they will move from the initially infected cells to the vascular tissue, and through the phloem move up and start invading a whole plant. So the virus gets inoculated in a cell, opens up, proteins are produced, the virus factory produces millions of copies of the virus. These copies get encapsulated again in the basketball type shape particle. The insects can move them further. In the meantime, the viruses move from cell to cell and invade the whole plant and cause disease symptoms. So some of the diseases that is bringing us together today are leaf roll and red blotch. And here are two typical, what I would personally refer to as typical virus symptoms on Cap Franc. Red blotch to your left, leaf roll to your right. So leaf roll, you have very often a uniform reddening of a leaf blade. Very often, the main veins remain green, and you have downward rolling of the edges of the leaf blade. In terms of red blotch, you have red blotches that occur, that will start develop from the edge of a leaf blade or from the center and eventually coalesce. So this is textbook talk. Let's go to reality. I'm going to invite you to participate in a little trivia with me this morning. Are you ready? Are you game? I know Larry's game, so let's do it. So I put up on the left, red blotch, leaf roll. Just to make your job easy, I'm going to show you only Cap Franc infected only either with red blotch or leaf roll. Okay? I'm going to ask you to guess what virus is causing this type of symptoms. So if you believe red blotch, raise your hand. If you believe leaf roll, raise your hand, please. Wow, you guys are good. How about this one? If you believe red blotch, raise your hand, please. If you believe leaf roll, raise your hand. You guys continue being super good. How about now? Red blotch, raise your hand. Leaf roll, raise your hand. Right? Leaf roll. How about this one? Red blotch, raise your hand. Leaf roll, raise your hand. Again. Red blotch. How about this one? Red blotch, raise your hand. A few. Leaf roll, raise your hand. Red blotch. Okay, let's move to wide-berried wine grape varieties. In terms of Chardonnay here, overall for red blotch or leaf roll, symptoms are less conspicuous as in red-berried varieties. And you see for red blotch these interveinal chlorosis that looks like a lot like nutritional deficiency. For leaf roll, you have an overall chlorosis of the canopy and downward curling. These are textbook pictures. How about the reality here of a Chardonnay that has been extensively netted because to prevent bird damage? Red blotch, raise your hand. Leaf roll, raise your hand. Come on, guys. It is red blotch. Wow, who would have guessed, right? I was in your shoes many years ago. Blindly, I would have bet many cases on red blotch, and I was way off. Anyway, the last one. I have a series of pictures here. Only one is red blotch. Others are due to leaf roll, physical damage to a vine, nutritional deficiencies, or insect damage. Okay, you had a chance to look at this panel. Red blotch A, raise your hand. One in the back. Red blotch B. Red blotch C. D. E. The jury is still out there. F. G. Okay. The group is split. H. I. J. The person in the back is the winner. A is the only red blotch infected sample shown here. Quite amazing, huh? B is leaf roll, C is leaf roll, D is crown gall. E, you see the little arrow at the bottom right there of the picture, is girdling due to a tie. You see a white arrow in the upper left corner. It's a broken shoot in F. G is mite damage, and the three others are nutritional deficiencies. Anyway, so more importantly than just foliar symptoms is obviously the effect on productivity and fruit quality. And if you can look at this comparative picture of Cap Franc, you can see that on the right-hand side, you recognize the leaf roll disease symptoms, but try to count the number of clusters. And as you can see, a depression in the number of clusters, so yield impact of leaf roll to the right. Same for red blotch, and this is Pinot Noir. You see the symptomatic disease vine to your right. Look at the compactness of the leaf clusters, and more so the coloring of the berries in terms of anthocyanin in the skins and so forth. So yes, leaf roll and red blotch are very damaging in terms of yield and quality of production. And we mentioned, just to follow up on Ken's presentation, that viruses are transmitted by different insects. And again, there is an intimate relationship between a virus and a vine, and so exists an intimate relationship between a virus and a given vector. And just to emphasize what Ken said, different viruses are transmitted by mealybugs and soft scales. They are not transmitted by any other insect species. Dagger nematodes are transmitted by fan leaf, and other related viruses are transmitted by dagger nematodes. And this is just depicting the type of insect vectors that are known to transmit viruses from vine to vine or from vineyard to vineyard. And just to illustrate, I know that Gerhard later on will provide you very valuable insights as to how leaf roll is spread in vineyard. Just want to take this opportunity and show you what we have done in terms of red blotch. And this is just an overview of an estate where there were a number of Cap Franc and Merlot in Napa County. And I was very interested in the Cap Franc in the middle there, because to the east side of the block, and you see the red dots there, I saw an aggregation of diseased vine. And at that time, I had believed, when this vineyard manager called my attention on this Cap Franc, I believe that the disease, red blotch, was emanating from the riparian area to the right-hand side there, and something was coming out from the woods and starting the infection. Well, I can tell you I was way off, because the infection started with bundles of planting material that were infected, and somehow all ended up at the edge of the vineyard there, close to the riparian area. Anyway, it's a 5-acre vineyard planted in 2008. The first symptoms were observed in the 4th leaf in 2012, and the virus was confirmed present. So I mapped that vineyard. So each cell represents a single vine. Red is symptomatic, so disease with red blotch, and white blank is asymptomatic. And we monitored with the students in the lab this vineyard over 5 years. And just for the sake of our conversation, today I'm going to split this distribution of infected vines over time throughout the whole vineyard, or throughout the delineated section to the right-hand side close to the riparian area there. And this delineation is just artifactual. It's just me coming with a computer and drawing a little square around that block there. So overall in 2014, again, that's in the 6th leaf, the overall vineyard had a 4% disease incidence. And in the sub-portion there, in the subsection of vineyard, the disease incidence to start with was 32%. Now buckle up, I'm going to walk you through this vineyard from 2014 to 2018. So the newly infected vines are in green here. 2016, they're in blue. 2017, they're in purple. And 2018, they're in peach. At the bottom, I forgot to mention that the graph represents a transect of the vineyard across rows in 5 vine panels. And basically what you see is obviously a differential dynamic of red blotch spread throughout the whole vineyard, and in the section that I artificially delineated, the section where initially there were a strong aggregation of infected vine. And this is a take-home message in terms of management. I will come back later on. It is very, very key. So again, throughout the whole vineyard, just to summarize this type of work, there is a 1 to 3% increase of red blotch. And you say, ooh, what's the big deal? I can live with 1 or 3% incidence in my vineyard, which is absolutely correct. However, when you look at what is happening at the sub-portion of the vineyard there, where aggregation started the whole epidemic, you have a 10 to 20% annual increase in the number of newly infected vines. So if I summarize, viruses can be very impactful on business. And I teamed up with ag economists many years ago and your farm advisors. And these are the numbers we came up in terms of impact of leaf roll and impact of red blotch. What we know is leaf roll and red blotch, like any other grape viruses, only infect grapes. What I mean by grapes, I mean grapes in production vineyards, grapes in foundation vineyards, and wild grapes. So Vitis californica, along riverbanks and so forth, are also infected with red blotch and leaf roll viruses. Once a vine is infected with a virus, there is no cure in a vineyard. Okay? The virus-infected vine will keep the virus until it dies on site or it is removed. And particularly, there is no chemical that can be used against a virus. And the reason being, going back to my little general statements about how viruses are obligate parasites, you can easily imagine if we were to develop a chemical that is effective against the virus, that chemical automatically would have a side effect on the plant, which obviously would not be desirable. Viruses, including red blotch and leaf roll, are graft transmissible, meaning they can infect either the sign material or the rootstock, and means that also they can be transmitted through top working. So if you top work an infected vine, the virus will move up from the roots into the scion. Okay? And this is illustrated here. And you remember red blotch on Chardonnay that I showed before, this picture on the left? So a grower didn't meet the expectations in terms of ripeness of a Cab Sauv in the Lodi area because of red blotch. And the vines were tested positive for red blotch. Unfortunately, the grower believed that if he doesn't meet the minimal requirement with a red, he might meet it with a less demanding varietal in terms of standards. So he top worked these infected vines with Pinot Grigio, and he told me, I solved the problem. I don't see reddening. Well, you would not see any reddening on a Pinot Grigio, but you can see the similarities in the symptoms, this interveinal chlorosis. So basically what had happened is from the stock, or what is left from the Cab Sauv, the virus moved up on the top worked vines, and now he is dealing with a Pinot Grigio infected, red blotch infected vineyard. On a bright note, red blotch and leaf roll, like any other grape virus, is not mechanically transmitted in vineyards. And I have to explain you what I mean by that. It means that hedging, pruning, harvesting, suckering, you name it, does not move a virus from an infected vine to a healthy vine, from an infected vineyard to a healthy vineyard. This being said, again to tie in with some of Ken's comments, mechanical activities in a vineyard can move the insect vectors that do carry the viruses from an infected vine to a healthy vine, from an infected vineyard to a healthy vineyard. Okay? And I'm often asked, what is the implication of insects in terms of movement of viruses, particularly with the red blotch, relative to the planting material? And I can tell you, based on my observations, my limited experience with red blotch, 90 to 95% of the presence of red blotch in a vineyard is explained by the poor selection of the planting material, not the insect vectors. What is challenging in terms of visual observations of virus presence based on disease symptoms, infection is in most cases latent in rootstocks, meaning that rootstocks can be infected, but they will not express any disease symptoms. We already talked about the role of insects in vineyard transmission, in virus transmissions in vineyard. So in terms of management, trying to put everything together, what have we learned? You talk to a virologist, he will tell you, and if you talk to most of the entomologists, they will tell you the same. Focus all your disease management efforts, the majority of it, into eliminating the virus inoculum in your vineyards. That's where you should put the emphasis. I know it's such a simple message, but you remember the disease spread over time for red blotch? I kept on telling the grower to eliminate that part of the vineyard. And obviously, he didn't listen to me. Why would someone listen to a French-American hybrid anyway, right? To start with. But can you imagine how much he would have saved in terms of the quality of the production if he just had removed that section out there? Anyway, that's his decision. And I'm fine with the decision, because a management decision is based on the incidence of the viruses, the distribution of the viruses, how this distribution might change over time within the business model of what you are carrying out. And if you decide to live with the viruses, that's perfectly fine to me, as long as you are aware of the consequences and the risks you're taking. Okay? So, in terms of management, scouting for leaf roll and red blotch right around grape picking time or right immediately after. Flag the vines that are suspicious. Make sure you test some to know whether you're dealing with a virus, right? Remember the different slides and the trivia we just played a few minutes ago? Know whether you're dealing with leaf roll, with red blotch. And if your disease incidence is roughly 25%, rogue. Eliminate individually infected vines. And particularly make sure you avoid the infected vines being aggregated. Otherwise, the disease will go rampant very, very quickly. If the disease incidence roughly is more than 25%, you are better off removing the entire vineyard and replanting. Now, obviously, there are many considerations. These are just a guideline. Doesn't mean that you have to apply this 25% threshold to every of your blocks throughout the whole estate that you're managing. Just a guideline to keep in mind, because there are price differences, price penalties, and you know best the history of your different blocks than we do. So now in terms of knocking down or reducing the populations of insect vectors, in terms of leaf roll, as Kent mentioned, I'm very supportive of the use of insecticides, particularly knowing how efficient some of the chemistries that you have been using are, and I agree that the timing of these applications are really key to knock down and the insect population with the aim of reducing the spread of leaf roll viruses. But again, keep in mind, eliminating the virus inoculum should be the major target of your management strategies. Okay? In terms of red blotch, I do not recommend the use of any insecticides. This is because the virus population, the insect vector population in a vineyard is not abundant. This vector does not like grapes. It does not reproduce on grapes. The three-cornered alfalfa hopper hates grapes, actually. So you would have a tough time knocking down the population if it's not residing within a vineyard. Okay? And the last take-home message is be careful and spend quality time selecting your planting material. Again, just to conclude, focus your efforts on eliminating the virus. And with no further ado, I thank you so much for your attention. I want to thank all the sponsors that contribute to my research. Thank you.
Leaf roll virus for us is probably the biggest threat that we face in the industry. I mean, more than labor, more than anything. It's a huge cost, and this meeting brought together it combined the foremost authorities on the subject in the industry globally, and the knowledge they provided was fantastic.
Professor Peterson has been studying leaf roll virus since 1990, really, right? He was invited to consult with New Zealand, in 2007, and now we have the pleasure of consulting with him to learn how to deal with leaf roll virus management in California. Please join me in welcoming Professor Peterson from South Africa.
Thanks, Stephanie, for that for the introduction. I don't really advertise the fact that I've been working on leaf roll since 1990. It's a little bit sad, and especially so if you look at how slow information actually gets generated. It's a massive privilege to stand here and talk to you and to share with you some of the experiences we've had in South Africa in trying to manage this disease. I think we had to. We had no option. The virus is so prevalent in South Africa. The vine mealybug has been there since the 1930s. It's absolutely ubiquitous. So it was really essential that we needed to look at controlling this disease. So my talk today is not so much on a nitty-gritty level. I think I really would like to get some of the concepts across, and the detail we can really discuss around question time. So, without a doubt, grapevine leaf roll is the most important virus disease in South Africa. We are very fortunate we don't have grapevine red blotch. It causes an impairment of the vascular tissue, and I really must thank Mark and both Kent for supporting information to my talk. It really will help a lot. So it causes impairment of the vascular tissue, which results in a number of undesirable properties. The berries don't color up that well, the bunch architecture gets affected, they take longer to ripe, they don't reach sugar levels, yields drop, and in a really recent, a very nice study about a year ago in South Africa, wines made in a very standard method, in other words, not compensating for deficiencies in juices and that, showed that with wine tasters, the country's top 30 wine tasters, showed that there's a definite sensory penalty that's paid for having virus-infected plants. So the other thing is the virus spreads quite rapidly in South Africa. We monitored about 80 vineyards in the late 1990s, early 2000s, and we could show that spread was about doubling every single year. And this is just one of those vineyards. You can see over years as it progresses. There is probably not a single vineyard in South Africa at age 8, 9, where a grower isn't controlling leaf roll, where the incident isn't at around 100%. It is really rife. And the result of this is that our vineyards need to be replaced every 20, 25 years, almost directly as a result of leaf roll. This, of course, has enormous economic consequences. The most important virus, and I'm glad Ken showed the slide that there are more leaf roll viruses, leaf roll associated virus than just type 3, but type 3 is far and away the most important one. And you have a similar situation here in California. This is really the target that we have to reduce. Mark also mentioned to you that there's no chemical that you can use to try and spray for the virus the way you would for a fungicide. So in the case of controlling viruses, we have to look at the epidemiology of the disease. Any of you who've done some modules in plant pathology would recognize this figure immediately. It's the disease triangle, and it basically tells you that a disease epidemic occurs when all of the components that I've got over here are together and you have a susceptible plant. So let's look at the individual components of this for leaf roll. Let's turn our attention first to the plant. Firstly, all the Vitis vinifera cultivars that I'm aware of are susceptible to type 3. I have not come across a single cultivar that hasn't been susceptible to it. It also affects those that don't show the symptom that well, as my two colleagues pointed out. There are only some cultivars amongst the white ones where you would actually see the curling of the leaf as well. Something like Chenin Blanc and Sauvignon Blanc don't really show symptoms ever. It's important that we realize as well that the virus affects the rootstocks, and these are of course other species of Vitis vinifera and crosses of them. Although having said that, our most recent work has shown that there's some interesting things happening around the rootstocks. They don't show any symptoms, but if you take a plant like this, typically the rootstock would show absolutely no symptoms, even though it's on a scion that shows very, very severe symptoms. If we test those examples, then in spite of that constant pressure from the inoculum at the top, some of the rootstocks don't get infected by the virus. About 2/3 of the plants that we tested had this incident. So we really don't quite know what's happening there, and this is a very exciting avenue for control that we need to pursue. If we turn our attention to the virus, and ladies and gentlemen, I cannot thank Mark and Kent enough for telling you this is the weak link in this chain. This is the weak link, the virus. The mealybug is a complex organism. It's difficult to control. The virus is pretty simple. That's where we need to focus our attention primarily. That doesn't mean that you cannot ignore the mealybug. You have to reduce its level, but this is where the focus has to be. As Mark mentioned, we are very lucky, and this is one of the weak links of this virus, is it doesn't spread by mechanical means. So sap on a harvester, sap on pruning shears, that won't transmit the virus. Secondly, and this is the single biggest weak link that this virus has, is the fact that it only infects Vitis species. It means we don't have to worry in our management. We don't have to worry about weeds. We don't have to worry about alternate hosts on the outside of a vineyard. All our effort can concentrate on Vitis. As Mark also showed, the virus can be vegetatively propagated. So if you take planting material from an infected plant, most of those cuttings will also be virus infected. And similarly, if you do grafting and one of the two graft components is infected, the other component will become infected. Turning our attention to the mealybug, and I anticipated Kent's great talk, so I haven't gone into any kind of detail, barring the transmission properties of mealybugs. And I'm kind of worried, but in a sense, it actually looks as if our biovar, or our variant of the vine mealybug, might be even more efficient at transmitting leaf roll than the one that you have over here. So Kent showed that a number of species of mealybugs and actually some scale insects can transmit type 3, and that, as in your case, vine mealybug is the biggest problem in South Africa. My colleague Kirsten Kruger showed that the efficiency that we find with our variant of vine mealybug is 70% efficient. So what Kirsten did is she took individual crawlers, put one onto, had them feed on an infected plant, and moved single ones to healthy vines. And in 70% of those instances, that new vine became infected. So they're 70% efficient at transmitting the virus. Another problem that we have that also looks a little bit different to your situation is that the vine mealybug in South Africa, the ones that we used for these experiments, can only need to feed for about 15 minutes to actually acquire the virus. And they can actually, by feeding another 15 minutes on a healthy plant, they can transmit the virus. So the numbers are even lower than those shown by Kent. And it's probably because of different biovars of the mealybug. This implies, as Kent also showed, that this virus is semi-persistent. It's a term that we use in virology. And that implies a number of things. It means that the virus is lost. It's only associated with the mouth parts of the virus, of the insect. So it's lost on molting. It doesn't replicate in the host, and it doesn't get passed on to the offspring of a host, of the insect. Now, mealybugs in general on their own aren't that motile. They need wind or other methods of dispersal, and this can be by the wind over short and long distances, by birds or ants, by man through some of our agronomic practices on implements and on workers. And so the epidemiology, to boil it down to its simplest level and taking it from the virus perspective, is quite simple. How does an infected plant actually become infected in your vineyard? And there's only two ways. The one is that it was either the planting material, that's the only one of the two ways in which that vine got infected, or secondly, the virus was introduced on a mealybug. That mealybug could only have fed on an infected grapevine. This, in its simplest form, is the epidemiology of this disease. Now, the weak link is the fact that this mealybug has to go and feed somewhere on an infected vine. If we can remove the infection, we can tolerate mealybugs in a vineyard at higher levels than we can while the virus is in the pathosystem. So, knowing all this information, in the early 2000s, a little bit earlier than that, we did spatiotemporal analyses of the way that this disease spreads. And this gives one fantastic clues about how to go ahead and control the virus. So we want to determine what are the most common means of spread. And the far and away the most common pattern we saw was this number of infected vines in a row and then movement over the rows. So if you follow the cartoon here, you'll see that was the infection in the first year, and you'll see there are already instances where there are 3-4 vines right next to each other that are infected, which would have happened before we started monitoring. And then as time goes by, you can see how the spread moves out, firstly along a row and then over a row. This occurred in just about every single vineyard that we monitored, and it gives rise to these runs of infected plants and ultimately foci of infection. This is spread from that one infected vine to its neighbors, and it's known as secondary spread. It's the spread that happens within a vineyard. And this could be through the mealybugs, through their own motility, being moved on by ants, carried on implements from one infected plant to the healthy plants right next to it, or on workers. The second pattern that's very common is edge effects or gradients of the disease over a vineyard. And in this block, the maths isn't that important, but this gradient, you could see clearly how the infection started from this lower portion and how it actually moves further on into the block. I also want to point out that this is at a perpendicular orientation to the row orientation, which would mean that this kind of spread is not associated with implements or man, but things like wind or birds. You can see how it moves further into the block and how secondary spread starts moving those single vines further within the block. And that gives you these typical edge effects. I'm sure you all see it in your vineyards. This happened in about 25% of instances where the row orientation between the infected block and the healthy block are the same. It happened in slightly lower numbers when the orientation was perpendicular to each other. But we also saw it where there was no obvious vineyard surrounding the block that we were monitoring. And this implies that there's a long distance component as well. The virus can be brought in on mealybugs over longer distances. And these, once again, are possible through implements, once again through workers, blown on the wind, blown on plant material. The third most common pattern we observed, and this would have been hidden in most of the instances. So it's only a few instances where we would be able to see this pattern in, and this is very early in the life of a vineyard, where you have individual infected plants. Very often these symptoms would come up within the first season or two, and that's generally quite a good indicator that it's through planting material, especially when it's only associated with specific rootstock scion combinations. As in this case, you can see the middle cultivar didn't have any virus, whereas the other three combinations had the virus. This could be spread through the planting material, and it can be both the scion or the rootstock. The last pattern we observed, and we actually at that stage just had one example of this, was a very complicated one. The grower had very infected vines on the entire area here since the 1980s. He removed them in total, and then in 1999 he only planted the orange portion at the top. He left the bottom portion, the green portion, fallow. That block almost immediately got leaf roll, to the extent that it probably was planting material. However, what he did is he removed that top portion after only one season, and then he planted the area that's in these red dashed lines. And you can see in this red dashed cultivar now that there's a much higher level of leaf roll infected plants than in the area that was fallow. So this gave us a really good indicator, circumstantial evidence, but good indicator that the leaf roll was coming through from the previous vineyard. And if you look at vineyards that are removed and they lie fallow for a year or two, you'll often find volunteer hosts coming up, sometimes even with symptoms, which implies that it's some of the scion material that's coming up, canes that were just in the ground and that have actually sprouted. So knowing these spreads, and I'm sure you have all of these patterns of spread in California, how do we go about controlling this disease? Well, we have to prevent each and every one of those means of spread happening. So the picture around epidemiology becomes a little bit more complicated. We have a plant that can be infected either through planting material or through varioliferous mealybugs. The mealybug must have fed somewhere on a grapevine. That grapevine could be amongst the peer plants in that same block. It can be volunteer hosts. It can be root remnants. It can be from vineyards very close to the the vineyard that you're establishing, but it can also be from vineyards quite far away. So let's turn our attention to each of these. Planting material. Now, we have a certification scheme, as you do, and in our certification scheme, every single clone that gets registered within the certification scheme has to pass through virus elimination to ensure that it is free of leaf roll, and then it is propagated initially in nuclear blocks, then into foundation blocks, mother blocks, and these are distributed to nurseries. The virus elimination process is quite complicated, but it's very, very effective. The plants are put in a chamber at about 30 degrees for 3 months, and then the absolute uppermost cells in the meristem are cut, and a plant is regenerated from those cells in tissue culture. This works extremely well for type 3, and those plants derived from that are known as our nuclear plants, and we have had no leaf roll infection in our nuclear plants over many, many years. The important thing to remember is that those plants are now only virus-free, leaf roll-free. They are still susceptible to the virus. They're not resistant to the virus. They can still be reinfected. And the minute you take them out into the field where there's an inoculum source, they will become reinfected. So one of the things that's done to prevent this is our foundation blocks, a small percentage of them, are taken completely outside of the grapevine production area. They planted out in the desert, and there's no other vineyards around them. Those plants are as close to being safe from leaf roll as any plant could be. These are very expensive vines. They're called 3-star rated material, and growers would pay a premium to actually plant material from them. But the large volumes that our industry needs, and you're a bigger industry than us, it's not possible to have your blocks isolated in that way. So in South Africa, the next level of blocks often get planted at commercial farms. The grower will use the berries, and the plant improvement organization will take the canes. So in that situation, there is going to be reinfection because there's a lot of leaf roll in the environment. So in the red cultivars, we have inspectors going through and culling out, droguing the infected plants. And then in the white cultivars, they're tested yearly and the infected plants are removed. Now, a very important principle is that leaf roll is a little bit like HIV in the sense that it has a latent period. A period in which you're not going to see symptoms, and a period in which doesn't matter if you use ELISA or PCR or even a more sensitive technique like LAMP, there is a period where the plant, even though it's infected, is going to test negative. So you cannot expect nurseries to actually provide you with plants that are going to be absolutely virus-free. So it's important that as a grower, you also do something about infected plant material. And in South Africa, what we promote is the use of this 3-star planting material where possible, and if not possible, to immediately establish your new vineyard and immediately treat the plants with a systemic insecticide. The idea behind that is that you would basically wait for that plant to show symptoms, wait for the virus to replicate to levels to show symptoms, but not to infect the plants around it. And then monitor these plants for a couple of seasons and then remove them. This works extremely effectively against plant material that's coming that's infected. This is an example of that. This was quite a horrific example. The grower, in his first season, had a 12% infection through planting material. Terrible, absolutely terrible. The fact that it showed symptoms so quickly showed us that it was due to planting material. But what we found very disturbing right in the beginning was those numbers of clusters that you see there. It suggested that in that one season, there was already spread. If that was true, we would not be able to control leaf roll. But luckily for us, that's not the case. We could trace this back to infection of the rootstock in that instance, and where the bundles of plants tended to have a number of plants from the same infected source, and they were being planted right next to each other. By treating that block with imidacloprid and roguing the plants, the next year the numbers are dramatically reduced from 548 that first season down to 27 the next season, 9 the season after that, 14 a little bit of a jump again, 17 bit of a jump, and then suddenly we were down to really single digits. When you have infection through planting material, the infection you see in front of you will more or less be the number that you need to remove in the end. Here we had a 12.2% infection in the beginning, and we only needed to remove 13.6% of the plants to get rid of this infection. So just slight little bit more than what we had seen in the beginning. So roguing to control infected planting material is incredibly effective. Okay, that's so much as now infection from the pear plants. This is far and away the method through which the most new infections take place. That secondary spread is the way that the virus really infects other plants. It's the most critical aspect of control that we need to address. If you look at this vineyard, and I want to draw your attention to one of the 3 arrows, and I'm only going to describe the top arrow. There was a single infected plant there in 2001. In 2002, there were 2 infected plants. 2003, there were 16 infected plants in that foci. The next year, 53. And then after that, they were coalescing, and we could no longer be sure that any infection plant was coming from a particular previous one. Had the grower controlled mealybug and had removed that single vine in that first season, he'd have protected this entire foci. That wouldn't have happened. So the principle really is to get into your vineyard as soon as possible and rogue. It's the least painful way of doing it. You're going to have to remove the least number of plants by doing it very early and constantly doing it, never skipping a season. So that you can do in vineyards that are relatively uninfected, and we have a user threshold of about 20%. Mark mentioned 25%. That number is very variable. If you see a vineyard where you've just got individual infected plants, 20%, 25% is fine. If you've got a vineyard where you're seeing lots of clumps, where there's already been a fair amount of secondary spread, you can be sure that what you're seeing is only a portion of the plants that are already infected, and that you're going to have to remove a huge amount in the end. So I just want to illustrate this. In this vineyard, in the first season, there were 509 infected plants with some aggregation. In the second season, 211. The year after that, 110. Then 30, 22, 12, and 10. Now, you can see we're managing the numbers down very nicely, but ultimately we needed to remove 481 vines from that block, more than the initial amount that was there. So literally, the quicker you can go in, and as long as there's not too much secondary spread, you can reduce the numbers. This is still a far better situation than had this block been had been left. Typically in South Africa, that vineyard with 509 infected plants initially would have become 1,000 the next year, 2,000 the year after, 4,000 the year after. It spreads very rapidly if you don't do anything. It's important to remove the vine totally, in its totality, as much of the roots as you can, and the entire top. We mark the plants in autumn, and we remove them in the winter when the soils are wet. When you have a high incidence, above about 20% in a vineyard, that's it. You can no longer control leaf roll in a vineyard like that, but you must control the mealybugs. Those vineyards can still remain as sources of infection to a block next door, which might have a very low percentage of the virus. Okay, volunteers, how do we deal with replanting a whole vineyard? Well, in the preceding old vineyard that you're about to take out, you need to, just after harvest, apply a systemic insecticide, imidacloprid. This is very counterintuitive to a grower because that's the block you're removing. But what we're trying to achieve with that is that the imidacloprid be taken up into the roots, so that when you remove that vine and any of the root remnants stay behind, that they wouldn't support mealybugs. Then we recommend a fallow period to get rid of any of the volunteer plants that come up. Now, there isn't a time limit or a time coupled to this fallow period. We're not saying 1 season, 2 seasons. It all depends on how effectively you remove that vine. If you've got light soils and you can actually remove the vine in total with very little roots left behind, you don't need a fallow period. If you have got heavy soils, lots of the roots are breaking off, you need to get those roots out of the ground, you need to plow them up over fallow season periods, and you need to get them out of the system. You don't have to get rid of all of them. You just have to ensure that you're reducing the risk. And in your new vineyard, you should keep on removing any volunteer plants from that previous block. How do we deal with when the infection comes from far, these typical disease gradients? Well, it's very important to control the mealybug in both adjoining and distant vineyards. We recommend that you work in the healthy vineyards before you move on to the infected ones, and where that's not possible, that implements get cleaned down just with a normal dilute soapy solution before entering the healthy vineyards. What if your neighbor doesn't control leaf roll? This situation does happen, and we really, both Kent and Mark stress this, and I can't stress it enough. If you can do control on an area-wide basis, the period between the industry being half clean and half infected will be shortened. The pain of dealing with that period will be lessened, because most of the complications in controlling this disease is in that period where there's both infected material and healthy blocks. And this is really, if your neighbor doesn't do it, release predators and parasitoids. They will help clean up your neighbor's vineyards as well. Some examples of leaf roll control. The first place they did it, and I was extremely lucky that this estate called Vergelegen, and I challenge you to say that. Vergelegen means situated far. It's an old estate, one of our really top-notch estates. It was established in 1700, and at that stage that was very, very far from Cape Town, hence the name situated far. It's arguably, it has the South Africa's top winemaker, so it was fantastic that these guys were interested in controlling leaf roll. And we were able to exploit some situations. They were in a phase where they wanted to expand on the vineyards, so they were removing citrus orchards and re-establishing vineyards in those places. So that was our first phase. And these were planted between 1999 and 2003, and it represented about 900 acres. Our initial infections in those blocks there was about 600 infected plants, which represented a 0.6% infection, very low, relatively low at a start of a vineyard. But remember, had we done nothing, those numbers would have doubled in every year after that. And we've calculated if nothing had been done, these blocks would have been 100% infected in about 9-10 years' time. However, we managed to reduce, using all the interventions I've mentioned, we've managed to reduce the infections to now 0.049. That's about 5 infected plants in 10,000. For the last 8 or 9 years, we've been getting roughly 30 infected plants on a yearly basis. We're not completely getting this disease eradicated. And I think the reason for that is this estate is on its own in controlling the leaf roll. Their neighbors are still not doing it. Phase 2 was a little bit more complicated. This was replacing old 100% infected vineyards. Here we had to do a fallow period. So initially they treated the plants with imidacloprid, removed the old vineyards, had a fallow period in which they removed all the remnants from the previous vineyard, and then we re-established new blocks. I've kept the scale of the graph the same as the phase 1, just to show you that in the meantime, planting material in our certification scheme who had also applied this control strategy had improved significantly. Now we only started with a 0.09% infection level, and we could manage that down to roughly 0.03. That is minute. It is the exception to find an infected vine in these red cultivars. That whole vineyard that you see there is Cabernet Sauvignon, and this is a typical site, maybe just 1 infected plant per season. Had we done nothing, this spread that I showed you earlier would have happened. Now, we were working in levels of complexity. So phase 3 was the most difficult. This is where we had to replace white cultivars with new vineyards. This was done in a much more phased approach, and probably a more realistic approach to most industries. So the blocks were taken out piecemeal and replaced with new vineyards. And it's still going on. We still haven't gotten all of the old vineyards out. What makes this phase very difficult is that you need laboratory tests to actually identify the infected plants. So what we did is we trained the cellar technician to perform ELISA tests. ELISA is a very robust technique. It can be taught to someone quite easily. It doesn't have a lot that can go wrong with it. And so they test on a yearly basis every single white cultivar. They have composites of 10 plants, roughly. It's actually 2 bays, but so between 8 and 10 plants. And then if a composite comes up positive, we test the individual plants. And the pattern's very similar to what we see in the red cultivars. In this case, you can see a clear gradient because there's a block right next to it that's infected. But controlling this works equally well. Roguing it works equally well. And we've actually maintained the level of infection to below 0.3%. 3 infected plants per roughly 1,000. This is in the industry wide. In our certification scheme, most of our foundation blocks are applying these techniques now. This is 335 such examples. This is a very complicated graph, so I just want to go through it quite slowly. This reflects the age of the vineyards, and it's the wrong way around. The oldest ones are at the origin of the graph, and the youngest ones further down. The incidence is on this axis, and I want to point out that we're looking at 0.5% as the maximum on this graph. There are only 4 vineyards that actually exceed this amount, those 4. Then the blocks are ranked by age, and within any age group, they're ranked by the percentage of leaf roll that's there. So looking at, say, this 9-year-old vineyards, and there's probably 130 in that cohort. The vast majority of them no longer have leaf roll. These other ones, the highest level is 1.38% in that block. So really dramatic control. Similarly with mother blocks, it's all pretty similar. There's 119 of them in this instance. The highest infection is 1%. All similar. Now, it's not just in South Africa where this control strategy is being done, and New Zealand has also done this, and even better than we did in South Africa. They decided they wanted to clean up a whole appellation. So up on the Northern Island, in the Napier Hastings area, there's an appellation called the Gimlet Gravels, mainly red cultivars, luckily. 30 growers, all 30 growers of that appellation decided they want to apply leaf roll control throughout the area. They mapped 100% of the 800 plus hectares. That I didn't convert that to acres, but it's massive. And they had a vision of doing this over a 4 or 5 year period. These are the numbers. So those blocks that were more than 20%, that usually we'd have said, okay, don't control leaf roll in that, they decided they're going to remove those blocks. So over 70 hectares in the first phase, that's 1,800 acres in 2010, were brought down to only 185 acres by 2015. In the vineyards that had less than 20%, the numbers were initially over 27,000 plants that were infected. By 2014, that was down to 9,000. I no longer see them on a yearly basis, so I don't have more recent information, but area-wide, this control has been incredibly effective. Once again, remember, had they done nothing, these numbers would have just climbed. So, at the very least, ensure that every new vineyard you establish, that you use these control strategies. It's going to differ. California, from what I've gathered from talks with a number of you, you have many more cultivars in Lodi area than we generally use in South Africa. So we're very lucky. Our main cultivars are Cab Sav, Merlot, Shiraz, and then Pinotage, and they're really easy to see the symptom on, so that we can really control a large number by just targeting those cultivars. I think that situation, you would have to adapt to the California situation. The important thing is it can be controlled. It's generally best when it's done area-wide. The greatest risk is in this period where there are many infected plants and you're still trying to establish new ones. The greatest need for mealybug control, the lowest levels you need, are in this transition phase. Once the inoculum level has been reduced, once the virus level is there, you can actually tolerate much higher levels of mealybugs in your industry. So really, it's a case of focusing to really try and get through this phase as quickly as possible. And then after that, to really make use of a more sustainable way of control through biological control, basically to ensure the sustainability of the entire process. In many ways, we're a victim of our success in South Africa. I'm having an incredibly hard time convincing growers who've been controlling mealybugs through insecticide means, who've reduced the numbers down to very, very low numbers, to now go over fully into biological control. This is this year's harvest from Vergelegen. The grower, we've got blocks that are now 19, 20 years old. The yield hasn't started dropping. The quality hasn't started dropping. The harvest can be done in a single go through All the berries are pretty much ripe at the same time. There's no color problems. Everybody is smiling in this regard. I want to really thank Stephanie and Lodi Winegrape Commission for inviting me to come talk to you. WineTech, who's been supporting my research since 1990. I really take my hat off to them. And then my own institution, Stellenbosch University. Thank you for your attention.
So next up we have Dr. James Stamp. He's going to be talking to us about planting material, and it's very nice that he agreed to come and speak with us today. We really appreciate it, and we look forward to learning from you. Thank you. Let's welcome James.
Thank you. Okay, thank you very much for inviting me here today. I feel very privileged to be here with such a great group of growers and experts in all their fields of relating to grapevines. I'm going to talk about grapevine planting material, and hopefully it makes sense and it's interesting. The goal of quality assurance for grapevine production, it's very basic. When you order grapevine plants, you want to make sure those grapevine plants are delivered to you, that they're high quality grapevine plants, that they're delivered in the correct amount, that your order is completely full when you get those grapevine plants, and that the delivery is on time. You don't want any surprises, so it's very important to be in good contact with the nursery through the production of your vines. So if there are any surprises, you'll know about it in advance so that you can actually make changes or figure out alternative approaches should your order be short. The vines, of course, should be virus test negative. That's a given, and the talks today we've heard are all about producing virus test negative vines. But of course, they should also be of good physical condition. And the physical condition of the vines is directly related to the pathogen load of those vines. So if you receive a batch of vines that have bad graft unions, poor root systems, poor callousing of the rootstock disbudding sites, the likelihood is that those vines will have high levels of fungal pathogens in them. So it's a good way of ensuring that you're getting plant material by actually looking at it and examining it before you plant it. Just a background for everybody who may not be aware of this. Today we're all talking about Protocol 2010 materials in California. This is a range of plant materials that have all been produced through meristem culture, as Gerhard discussed in the South African situation. The CDFA provides vine material production guidelines, and these are documented under the Grapevine Nursery Certification Program. Foundation Plant Services cleans, verifies trueness to type, and establishes foundation block vines in the foundation block vineyard. This vineyard now is called the Russell Ranch Vineyard, and they plant 2 vines per selection in that vineyard. This vineyard is located between Winters and Davis up in near Sacramento. So these 2 vines, whether it be Cabernet Sauvignon or Clone 47 or Sauvignon Blanc 01, these 2 vines in Davis are the source of all the plant materials that you receive from your nursery. So a nursery would request cuttings from those 2 vines. They'd get those cuttings, and they'd propagate those cuttings and establish their own increase block, which is basically a mother block, but it's an increase block of foundation-derived material. And they would plant those increase block vines, rootstocks and scions, and when they were big enough, they would harvest buds and rootstock cuttings from those increase block vines to graft to produce your order. So that's the origin of all the plant materials that you get, generally speaking. And if you have a choice, it's best to order Protocol 2010 materials because the likelihood is they're cleaner, they have less virus, and they're also newer blocks, so they're also less likely to have have heavy loads of fungal contamination. Propagation and quality assurance, these are some of the essential steps. Nurseries should be doing this, and you should do this as a concerned grower because we all know what a massive investment it is to plant a vineyard and have to take it out. So you want to be involved or asking questions about all the steps of the propagation process. This is what we do with my consulting business. So the first thing to do is to select a nursery that you feel comfortable with, and I'll mention some pointers in a few moments. So select a nursery that you like or you feel is a good nursery, but the most important thing is increased block selection within those nurseries. You want to work with increased blocks that look healthy. And increased blocks can vary from a few rows to many acres. So it's important to look at the increased blocks, test and tag the increased blocks in late fall. The best time to examine these vines is when the symptoms of the virus are most obvious, and this is basically late October to November, depending on the location of the vineyard. It's important to walk both sides of the rows of the increased block. It's actually quite interesting to see how vines can look differently when the sun is shining toward you or shining behind you. One nutritional, in one direction it may look like a nutritional problem, in the other direction it might just look like a leaf roll or red blotch. So it's very important to walk both sides of the increased blocks with your partner. If the increased block doesn't look good, walk away from it. Find another increased block. Find some other rows of contiguous vines that look healthy. And once you've found vines that look healthy, then you can test them. The virus titer is very variable, so you want to at least test 2 pieces of tissue per vine, given that you have a cordon vine, so you can capture the variability. And again, you want to test all of the vines that you're going to be using to produce the propagation system and the grafting that you need. Once you've tested these increased blocks, you want to keep sight of them. So you want to make sure that the materials that you tested are actually harvested, and go to the grafting to make sure those tags that you put on those vines are actually appearing at grafting time. And then you want to be in touch with the nursery to make sure that the callousing is successful, because you can know within 3 or 4 weeks of grafting whether your order is going to be filled or not. So it's always a good idea to talk to the nursery 3 or 4 weeks after grafting, or 5 weeks, and ask them did they pot up the plants that were calloused, and how many vines did they pot up. And then look at the vines during the production cycle. So if it's a greenhouse production, if you're looking at green vines, you want to examine the vines in the greenhouse roughly halfway through the production cycle, and it roughly takes 20 weeks to produce a green vine. So around 10 weeks, go and look at the vines to make sure they're there, make sure they look healthy. You might also see at this point in time crown gall, which would then be very important to see that. Same with the field situation. Look at the vines in the nursery row. Again, if you see instances of poor take in the greenhouse or the field, you can see this very early on after grafting. You may have the opportunity to regraft if necessary and address the situation, which will obviously impact you later on if the vines aren't there. And then ultimately, you want to be involved, or at least ask questions about how they select and grade and test the finished product. If it's a green vine product, generally speaking, the vines will be clean because they will not have been in the external environment after doing your initial upfront testing of the increased block materials. However, if you're working with dormant bare root materials, it's a good idea to look at those bare root materials in the field in October to see if you see symptoms of disease, and you can see symptoms then. And also to screen them at that point for red blotch and leaf roll 3 to see if perhaps they've become contaminated in the nursery row. How to select a nursery. I think the biggest thing, apart from having a good location and having some experience, hopefully, of this nursery and their increased blocks, is communication. Like everything we do in this business, it's all about problem solving and communication. So if you have a nursery that will not call you back in a timely fashion, within 24 hours, it's difficult to actually move forward and make plans. So you need to have excellent communication and transparency. You want to have regular updates from the nursery as to the vine production status. So call them up if they don't call you up. The nursery should have Protocol 2010 increased blocks that are distant from commercial vineyards. Commercial vineyards, they could be wine vineyards or table grape vineyards, and they're often associated with mealybugs, so it's better to work with nurseries where the increased blocks are not close to commercial productions. The nursery should have an in-house virus test program. They may talk to you about this. Ask the question of how often do they test their increased blocks, what viruses do they test them for, and what percentage of the increased block vines are tested. So that's very important information. You want to take a look at that before you place an order to see what the history of those increased block vines look like before you do any further work. Does the nursery have a mealybug control and exclusion program? And do they have modern sanitary facilities? Factors affecting grapevine performance and longevity. As I mentioned, the physical condition of a grapevine plant is really tells a story about the internal condition of the grapevine plant. You won't get good root systems, good graft unions, good healing of the rootstock, the spudding site, if there's a high level of fungal pathogens within those vines. Given the fact that we've done the due diligence and that you've selected materials that are to graft that are virus test negative, the next step is to ensure those materials look healthy in terms of carbohydrate reserves and wood to pith ratio. But when it comes to examining the finished product, if the finished product doesn't look strong, then you want to walk away from those vines. And there's various ways of looking at those vines, and I'll discuss that in a moment. But fungal pathogens, bacterial pathogens, and viruses all affect the physical condition of the finished product. And so if you have a weak finished product, then of course mechanical, biotic, and viticultural practices will all impinge on that plant. So if you accidentally J-root the vines, or if you insufficiently irrigate them, if you have a weak vine because it has high fungal pathogen loads, the likelihood is that vine will succumb to disease and to young vine decline pathogens more rapidly than a strong vine. Virus agents of greatest concern, if you do nothing else in terms of planting materials in California, you should test for red blotch and grapevine leaf roll 3. There are other viruses of concerns, but those are the two that you must test for. It doesn't make any sense to plant grapevine plants in California without doing that minimal amount of testing. And those are the two viruses that are most likely to be there, and those are the two viruses that are going to have the biggest impact on your bottom line. We have found pathogens in Protocol 2010 increase blocks in the last 2 years. We found leaf roll 3 in a Repair Gloire increase block, and Pinot Gris virus in 1616 increase block, and grapevine Pinot Gris virus and grapevine yellow speckled viroid in a Cabernet 47 increase block. Agrobacterium is widely prevalent in all increase blocks and also finished product. And also, it's likely that the plants you get from a nursery program will contain fungal pathogens. Interestingly enough, this table of information was released from UC Davis FPS at the annual meeting in December. And at this point in time, they recognized that 21 accessions of plant materials at Foundation Plant Services, the source of all the plant materials that go to the nurseries, had red blotch. So it's a very sort of devastating information, I think, for the industry, that our basic source material, our foundation material, is contaminated with red blotch. And the question is, what will happen with that material? Will this disease spread? We'll find out in this coming few months. Red blotch, Mark did a very good questionnaire about what red blotch looks like. Again, in reality, red blotch and leaf roll 3, in my opinion, look very, very similar. It's very hard to distinguish them, especially when the virus and the disease has been present for a few years, or then the symptoms are basically indistinguishable. This photograph shows a young infection where it does look more like red blotch. This table here shows the data from the work we did between November 2012, when the test for red blotch was first available, through to the middle of 2014. Most of these are CDFA certified increase blocks, and we tested these over that 1.5 year period. And you can just see the high level of contamination of increase blocks, either with red blotch or leaf roll 3 during that time period. If you look at some of these combinations, such as NTAV, Cabernet 15, there were only 3 NTAV blocks at the time, and 2 of them had red blotch. The same with Cabernet 169, there were only 3 NTAV blocks at the time, and 2 of them had red blotch. And then clones such as Cabernet 7 and Sauvignon Blanco 1, it was very, very difficult to get plant material either without leaf roll 3 or with red blotch. This next photograph shows the result of planting a vineyard with red blotch in Rutherford back in 2012. The top row shows rows of vines that were virus-free, and those vines were the fruit was harvested at 28 degrees Brix. The fruit went through complete ripening, and the winery realized an FOB gross wine revenue of $90,000 per acre in their premium wine production program. However, the vines that were positive for red blotch were harvested at 25 degrees Brix. They had impaired ripening of the seed components, And that fruit fetched $7,500 in the bulk market. So a massive impact. Red blotch can have a massive impact. Sampling strategies. The best way to sample for producing grapevines free of viruses is to sample before you graft them. You get a much better bang for your buck. If you, so a Cabernet 7 or a Sauvignon Blanc or one increased block vine could have 300 or 400 buds on it. So if you test one vine, you can be testing potentially 300 or 400 subsequent vines that are propagated from that. Most nurseries graft at maybe 1.5 to 2 times overage. So if you have a Cabernet Sauvignon vine with 400 buds, you might be testing, at the end of the day, 200 finished product. So it's very important to test up front if you can. And if you're going with the green vine production system, the likelihood is it will not become contaminated during the greenhouse process. As I mentioned, visually evaluate and test and tag every increased block vine as late as possible while the leaves are attached. And in terms of selection of materials, you want to collect woody materials for most of the viruses, although you can also use green materials for red blotch and fan leaf virus. And if you're working with a nursery where you believe they have a crown gall problem, some nurseries have more problems with the crown gall than others, you could potentially go ahead and screen your samples for crown gall, Agrobacterium vitis tumorigenic strain, before you test for viruses, which could save you a lot of money. And if you're also working, for example, with your own budwood selection, if you like that, you could pre-screen that for red blotch and leaf roll 3. And then if it tests negative, go ahead and do other tests. So again, that could be very economically beneficial to you. Sampling strategies post-grafting. If you end up buying some vines from a nursery, you've had no interaction with these vines beforehand. If you can look at those vines in the nursery row during the fall before they get harvested, first you'll see if the vines are there. You'll see if they look healthy. You'll see if it's the right clone, right variety, and you'll see if it looks healthy in terms of development of red blotch and leaf roll symptoms in the nursery row. And you can see symptoms in the nursery row. If you're looking at a finished product that's already been harvested, use statistically significant methods to test that finished product again for at least red blotch and leaf roll 3. An increased block can vary from a few rows to acres and acres. This is just a diagram showing that in this particular increased block, there are 2 rows of Sauvignon Blanc and 3 rows of Durifo 3. So potentially you might a nursery might remove the Sauvignon Blanc 1 increased block, but leave the Durifo 3 block there. But they're right adjacent to each other, so the chances are the Durif could also be contaminated. This is a photograph of what a typical rootstock increased block looks like. It's basically a mess of grapevine plants, canes. The canes are all growing on the floor, they're all intertwined with each other. It's very difficult to get out there and test them, so we always test the rootstock increased blocks when the leaves have fallen. And this is an example of a defoliated increased block. You can see some yellow tags there on the vines. These vines are being tested and getting ready for harvest. This is a photograph showing you an increased block of a sign increased block. This is really the ideal time to test a sign increased block. The leaves are just about to fall off. This is a red variety. You can see some red leafing on the left-hand side of the screen. That's mechanical damage. But you can see it's a beautiful looking vineyard, and if you walk down that vineyard with your colleague and look for sick vines, avoid the block if there are sick vines, but tag all the vines for testing. We tag the end of the row, and then we put tags on usable cuttings so we can see those tags coming through when the cuttings are harvested and when they get grafted. So we know those materials have been tested. And here is an example of going through and testing a sign increased block after we've already visually evaluated when the leaves have fallen. In terms of red blotch detection, we've talked about what the symptoms of white varieties look like. It's very, very difficult to determine if white varieties have red blotch. This is a Chardonnay 4 increased block at the beginning of November. It looks perfectly healthy. A month later, the 2 dark green vines down the row were red blotch contaminated. So I see in increased blocks with Chardonnay that the leaves, contaminated vines tend to hold their leaves longer than non-contaminated vines. And in this case, the leaves sort of turn green bronzy. With red varieties, I see, from my experience, I see the development of leaf roll 3 symptoms in advance of red blotch symptoms. So this is a Cabernet 412 block, an Entave block that looked very healthy at the end of September. However, in the beginning of November, it looked terrible, and it was contaminated with red blotch. So if we'd looked at this increased block in late September, October, we wouldn't have seen this. So again, everything's about timing and looking at the vines at the right time. Fungal and bacterial pathogens are still of great concern, and that's what really got me involved in this business nearly 20 years ago, the concern for young vine decline. So the pathogens associated with young vine decline are Phaeomoniella, Phaeoacremonium, and cylindrocarpon. These are opportunistic pathogens. They're pretty much in most of the nursery stock you buy today, whether it's from any nursery, it's probably going to have some level of fungal pathogens. That's again why examining of the physical condition is so important, because basically, if you test them, it's probably going to have the pathogen. But if it's a strong plant with good graft union, good root systems, you know it must have a low load. Otherwise, it wouldn't be so strong. Pathogens prevent propagation wound healing in nursery stock. So standard propagation methods mean that most new vines are contaminated with opportunistic fungal pathogens. Opportunistic means that if the vine is stressed, they will make the plant sick. So again, if the vine is strong, it's less likely the vine will be sick. These pathogens affect the graft union and propagation wound healing and root development, and can cause the development of lesions at the graft union and rootstock base. Not only are the vines stronger if they have lower pathogen load, vines that are contaminated with disease show internal symptoms that are very characteristic. So on the left is a very healthy looking rootstock shaft. This is a 1-year-old dormant rootstock rooting. You should cut your vines in half. Obviously you can't plant them, that's the good news. That was a joke. Okay. So you should cut at least some vines in half to see what they look like. They should look like the image on the left. If they look like the image on the right, halfway between the base of the vine and the top of the vine, you should reject vines that look like that. That discoloration, that gumming, and those exudates are a result of the vine trying to cut off the movement of the pathogen through the rest of the plant, causing the typical symptoms of young vine decline. How to identify low pathogen, load healthy vines. The percent take in the field or in the greenhouse is indicative of the general health status of the lot. So if there are very few vines there, then probably the vines that remain aren't very good. Careful external and internal examination is essential. And dormant bare root vines should withstand moderate flexing. So if you take a dormant vine product and you bend it a couple of ways, if it breaks, again, you can't plant it, but it will tell you if there's something wrong with that vine. So here's an example of a vine that was bent. It didn't have a great root system to begin with, but you can see this halfway along there, you can see the crack in the trunk at the top part. And so I scraped off the bark after I cracked it so you could see how much of that vine was not there. It did have an inferior root system, so again, it's very important to select dormant product with complete root systems, because that way you know there will not be a rootstock shaft there, a rootstock shaft lesion there. Product, green versus dormants. Growers like different types of products, but the big advantage of green vines is they haven't been grown outside in the environment, which is quite important considering the spread of red blotch and leaf roll 3. If you're taking vines off the shelf, spec vines, you should test for red blotch and leaf roll 3, and you should evaluate the condition of the vines for fungal and pathogen load through the examinations I described. Last couple of slides here. I think sometimes we forget how important grapevine plants are to all of us. We ask a lot from them. This data I have here is from Napa, so I probably doesn't really apply to Lodi, or maybe I apologize for that. But I talked with one of my friends, and he said that he calculated that the net per vine fruit value from a 20-year-old Cabernet, Napa Cabernet Sauvignon vine at $10.56 pounds per vine was 572 vines. So one vine gave him $572 worth of fruit. The wine from that vine equated to $3,233 worth of wine from one vine over a 20-year period in Napa. So it's a huge amount of investment, and it's a really valuable product. So going forward, to me, it seems that we need to be having a discussion about how we not only get rid of viruses and keep viruses out of the vineyard, but how do we produce grapevine plants that are resistant to viruses. And with the advent of gene editing technologies, CRISPR, I'm sure you've heard about it, we do now have tools that will allow us to genetically modify grapevine plants so that we leave no trace of any foreign genes in those grapevine plants. And so my interest is to see that, and I talked with Erin about this the other day, is that, 10 years from now, are we still going to be here worrying about how to plant grapevine plants, or are we going to do something that's going to allow us to have grapevine plants that are resistant to leaf roll and red blotch, so we can actually avoid some of this discussion? So I think what we need to do is to engage consumers in a conversation about genetically improved vines, what are the pros and the cons, and how it could be advantageous to the industry in general. And just the final slide, what do you want to spend $3.50 on, a Starbucks or a vine? A big difference in terms of what you get from it. So my sort of point is that grapevine plants are undervalued. They're very, very important to the whole industry. But people don't like to pay more than $3.50 for a grapevine plant, but it's basically the basis of everything we do, and it's very, very, very important. I'd like to start by acknowledging Stephanie's awesome organizational events today, and thank you for inviting me. It's really a real privilege to be here with you today. And I thank my staff. I also want to thank the nurseries that work with us, and thank you for your transparency, nurseries, and our clients too. And thank you very much.
My favorite thing today as a new grower and new PCA, learning management, best management practices, and also when you have a young vineyard, keeping it clean and really getting after it, and learning also insecticides and rotations to really get after it. I think those are the biggest takeaways for me.
Aaron Lang is going to come up now, and if all of the panelists would come up, we're going to do a question and answer panel. If I can get some of the virus focus group members to collect index cards with questions around from the tables. Hopefully some of you guys have written some questions down. We all know that Aaron has a lot of good questions on his own, but we want to hear from everybody too. And we're also inviting Chris Storm up as a grower. Chris was one of the first people to really start doing virus management, Chris and Charlie and Aaron. And he's got a lot of practical advice, not only just in Lodi, but also in other coasts. And people in Washington State that talk to me about viruses, they say, Stephanie, we really started testing for viruses in nursery material and other material because Chris Storm told us to. So he knows what he's talking about. So without further ado, I am going to pass it over to Aaron for our Q&A panel, but please smile so I can take your photo. Thank you.
Good morning, everyone. So I'm here just to facilitate your questions, and I really encourage you while we have this panel of experts here today to please, ask away, because this is our opportunity. I'm going to start with a few questions that I have here in my hand, and then I think other folks are going to bring theirs up, and I'll try not to repeat too many. So this is for Dr. Peterson. After roguing, how soon do you replant, and is there a point where you would no longer replant that vine?
That's a more difficult question than it sounds. Replanting in young vineyards is very successful. So in those instances, most of our growers will actually replant immediately. A small percentage of those plants do become infected again, but it's a relatively small percentage.
Do you think that infection comes from vine mealybug being harbored in the roots below the system that came from the previously rogued vine, or do you believe it's from the neighboring vines coming in?
We don't know, but it's potentially both of those. In an older vineyard, it becomes really, really difficult to replant. So the idea is really, when you do start a new vineyard, try to get the infected vines out as quickly as possible. That allows you to do a replant as well. The older your vineyard gets, the more difficult it becomes.
Great, thank you very much. You're going to be on the spot again here, but I encourage, if any other panel member here has something to add to the questions, please jump in. But another one for Dr. Peterson here. When can you effectively see symptoms and rogue in a newly planted vineyard? I think Dr. Stamp may have answered that, saying that he saw some infection even at the nursery level. But are they apparent, these symptoms, at year one for the main varieties, Cab, Merlot, Chardonnay?
If the infection is due to planting material, then there's a difference in whether you will see it in the first season, whether it was through the rootstock that was infected or through the sign that was infected. So there's already a difference. And then Kent actually showed some nice data around when a mealybug actually infects it. You showed more than a year later.
Yeah, so you can have infections come on later. We did work with Monica Cooper and John in Napa, trying to find out how you pull these roots out, what you can do, and I was visiting in New Zealand as well, where they showed live mealybugs 2 years after a vine was pulled out, still living on the ground. The issue is that it's very difficult to use anything to kill the roots, so we tried putting Roundup on it, we tried painting it, shooting it, every different type of method to kill those roots. Mike McHenry has tried fumigation. It's just really hard to get anything effective more than about a foot and a half down below the soil. And we know if you've got ants, and we know you do have ants, that the ants will take the mealybugs to where the food is, and that's on the roots left in the ground. And I also think that it's harder here to go for 2 years fallow than it might be in South Africa.
Just one additional comment on the timing of symptom development and when they become apparent. And in your replants, my take is usually if you see leaf roll symptoms in year 1, the first leaf, it is highly likely the cyan buds that were infected. It's highly unlikely that the infection results from mealybug transmission. However, if it takes longer, if it takes the second, third, or fourth leaf, then you have two options. Either you're dealing with mealybug transmission, or the source of the infection is the rootstock, because it takes tremendous time for the virus to translocate from a rootstock into the cyan. Just something to keep in mind.
Okay, I'll just agree that we have seen leaf roll and red blotch contaminated vines in the nursery row on several occasions. Within about, the vines in the nursery row are generally planted in May or June, and so we've seen symptoms in October, November, which again would indicate, as Mark's saying, there would be a cyan contamination versus a rootstock contamination.
Okay, this question is a practical one about removing disease material from the vineyard. It's a two-part question. One, during the fallow period or before the roguing, is it effective to use glyphosate or another systemic herbicide to try to kill roots and to kill it versus physically removing the roots? And two, once you've removed that infected material, is chipping diseased material sufficient to remove the inoculum of leaf roll and red blotch, or is burning necessary?
The first part is we way back we did recommend herbicides as a means of trying to kill the vine, but we couldn't find a single herbicide that actually kills it all the way down to the roots. Exactly what Ken said. So we no longer actually use herbicides to do that. Remember, when you're talking about a virus, it needs a living plant material. It doesn't create spores. So in fact, burning or chipping it is not that important from the virus perspective.
Okay, great, thank you. This is an interesting question. Are there any plants, hedgerows, so on and so forth, that a grower can use as a border around a newly planted block to prevent the spread of vectors, possibly coming from neighbors?
So they actually try this to some extent in New Zealand, because in New Zealand they've got smaller blocks typically, and they've got these wonderful hedges. They go up about 20 feet. The idea is that the mealybug will be blown by the wind. If it's a wall, like one of these walls, what happens is that the wind goes up and over and just carries the mealybug up and over and actually pulls it back into the vine on the other side. So the idea with the hedgerows is that the wind would blow through it, the mealybugs would land on a plant that they don't survive on, and they try to feed and eventually die. I've not seen data on how effective that is. What Von Bell had in that area is he put up cards in between infected and uninfected vineyards and was finding a tremendous number of crawlers being blown by the wind to the level of 100 per little yellow sticky card per week. So the hedgerow might be effective, but again, it's one more thing for growers here to plant, to maintain, to worry about, whereas insecticide application, something like that, might be easier. In terms of cover cropping within the vineyard, this is something we've gone back and forth on because for years we tested and looked at cover crops in their role to increase beneficial insects. And we know that especially the obscure mealybug can survive on many of the covers and weeds that you put into the vineyard. The vine mealybug can feed on it and survive. I don't think it can reproduce on all of them, but it gives them a kind of a bridge for survival. So, no real answer there.
Are there any particular cover crop species that we should be avoiding, maybe in the case of alfalfa leafhopper? So should we be moving away from broadleafs and more towards grasses in order to reduce the habitat for certain vectors like alfalfa leafhopper, which doesn't prefer vines necessarily?
Yeah, so we had this discussion just an hour ago. I'm still not convinced that it's easy to control Tika to reduce the spread of red blotch. If it's in your area, there's a lot of it. We definitely are looking right now at the legumes in the row middles and trying to manipulate those numbers to control it. So you've got a question of having nothing in the vine rows, having it bare soil, and that probably will reduce the amount of Tika going through.
I'm sorry, Tika?
Three-cornered alfalfa.
Thank you.
Hopper, sorry. So I can't really give a recommendation yet on the use of trap crops for control of this pest, but it's something Houston Wilson at Riverside and I are looking at. Frank Salem's lab is looking as well. I'm going to pass this on to Mark because.
Because is the answer. The question is still pending actually, but I can share with you that the heavily infested vineyard sites in Napa County, we monitored cover crops, particularly legumes on which the hopper thrives and can reproduce to see whether they carry the virus. That would have indicated eventually a connection between the cover crops, legumes serving as preferred hosts for the hopper and bridging it with the infected vines in terms of their role in the epidemiology of the disease. And we tested over 500 legume samples over the years and a few other plant species from these heavily infected red blotch vineyard blocks, and we have yet to find a single sample that tests positive for red blotch. So in other words, right now, if you ask me what my recommendation is, continue doing what you're doing in terms of cover crops. This means that maybe next week or in a month down the road, once our knowledge evolves, we might have to change our recommendations. But as of today, there is nothing to be worried about cover crops being actively involved in the epidemiology of red blotch. Again, bottom line, get rid of the inoculum. All right, in case you didn't hear me.
This is an interesting one, especially from a grower's perspective or a nurseryman. What is the acceptable standard for leaf roll 3 or red blotch virus infection in commercial nursery stock? We all know that nurseries can't guarantee that they're delivering clean stock. Is there a standard or an acceptable percentage that's allowable when you're purchasing commercial nursery stock?
Well, for me, I don't think there is. I mean, we work very hard to make sure that the clients that work with us get vines that don't have virus, like red blotch, leaf roll 3, and the whole panel. We test for the whole panel of viruses. We have different testing panels for Protocol 2010 materials, non-certified and certified. But for me, from my perspective, I don't want my clients to get any virus. And so there is a way of doing this. If you have a production program where you test all your increased block vines and you maintain them in a clean environment and you have your foundation plant materials maybe in a clean environment or away from any other commercial vineyards, I think it's quite possible to do that. In terms of acceptable standards, if it's a red variety, well, maybe you can rogue it out fairly easily. But if it's a white variety, I think it's going to be very lucky that you'll see it in the first 2 or 3 years, and by that time it could be transmitted. So I think we need to hold the nurseries to a very high standard. They need to be providing us with vines and figure out how to do it, provide us with vines that don't have virus. And I know it's doable because we do it frequently for our clients. It's just a question of, can the nurseries put the emphasis, can the nursery put the resources into that? And again, if we're talking about a plant that costs $3.50, it's basically nothing, you know, I think. I mean, okay, it's a cup of coffee versus $3.50. What if the plant was $7? What if we had a plant that was $7? What would that give us in terms of a better feeling for whether or not it was a virus test negative vine? Yeah.
A $7 vine would give me a heart attack. I agree with most of what you said, but for some of us who have clients or bankers that require revenue after a certain amount of time after you've pulled a vineyard, you need to get a vine in the ground. So it kind of comes down to the relationship you have with your nursery. Make sure that you have a relationship with your nursery and it's not just a phone call, order the vines, and that's it. But if you do have something that, you know, a low level of testing positive in the samples you've received, or once the vine is planted, that relationship is going to really turn out. You're going to want to talk about what percentage of replants they're going to provide, and for how long. Are they going to participate in the sampling with you? Are they going to share the cost of that kind of stuff? So each nursery is an individual company, each grower is an individual grower. You can set up your own relationship, but I think we have to accept for large plantings that perfection is not entirely possible. That's just the reality of the state of our industry. So knowing that, build into your budgets a certain percentage of replants for the first 10 years. 2% of 2% of your budget, or 2% of the vines replanting, no matter what, but whatever it is. But practically speaking, and I don't want to negate anything you said, here in Lodi, I just don't think we can say 100% is expected.
Please.
Mark,
Mark?
thank you. You're asking what would be a good number in terms of the incidence. So just to make sure and consistent with my message, the number I recommend is 0. No tolerance whatsoever. But acknowledging all the comments that have been made, this is a lofty goal, right? But we should shoot for that goal, although it's a lofty one as we speak. And I know that everyone is doing its due diligence to try to reach that goal. Will it happen tomorrow? Will it happen next week? Will it happen in a year? I do not know. But if we work collectively towards achieving that goal, I think we'll be all better off, and the industry collectively will be better off. Now, what I'd like to say now is those who will drive that mechanism, that program to reach the 0 tolerance is each of you in this room. You are the drivers of the program. If you are not asking for extensively virus-tested material, you will not get it. You are the drivers. You are the ones who will help all of us reach that goal.
Thank you. Are there, for the growers of the panel and those who have experience in the growing of vineyards with a high incidence of infection, I'm talking where roving's not possible but not economically feasible to remove the vineyard, are there any good strategies out there to reduce the negative effects of leaf roll 3, i.e. production and phenolics, color, maturity? Is there lots of people out there selling snake oil to try to get us to buy, saying it's going to help mature that crop even with a leaf roll infection? Is there any practical experience that says that something out there works?
A bulldozer. That's the only thing I've been able to figure out. I'm serious. We did a very robust study, replicated trial with many different sprayable formulations, fertilizer, and nothing works. Even dropping the tonnage down to 4 tons to the acre, not economically viable here, didn't work.
So what I'll add to that is that this came up in Sonoma because there were some old Zinfandels that were just not going to come out, and the grower had close to 100% infectivity, and my suggestion was, well, that's the block where you should be worried about mealybugs. That's the block you should be treating, because every mealybug leaving that block is infected. So don't give up on the mealybug control. If you're planting something clean next to it, worry about the block that's 100% infected with the virus.
I support that fully.
So basically, just to add up, the message is based on the fact that a vine that is virus infected, whether leaf roll or red blotch or whatever you name it, is a stressed vine by definition, right? So the more you do in order to mitigate the effect of leaf roll in favor of the vine and favoring the growth of the vine, the better off the vine will be able to defend itself against the virus infection. And we have to keep in mind that there is no silver bullet out there, because the severity of any virus symptom with a leaf roll, red blotch, and any other depends on the clone that you choose on top of a rootstock in a given environment, in a vineyard managed in a certain way. So something might be working to mitigate the impact of leaf rot in one vineyard, might not be translatable into similar positive results in another vineyard. But bottom line, try to boost the growth and the vigor of the vines so that it has a chance to better fight the virus infection.
Thank you.
I just want to say one other thing, and I think, as Gerhard mentioned in his presentation, I think it's very good if you can communicate with your neighbors so that you could potentially have a small area or a local area of replanting, just as you did in the, where was that? Yeah, yes, exactly. Right. And you know, it's amazing how many times we see new blocks getting planted, new clean blocks getting planted, say in Rutherford or Oakville, and they're completely surrounded by dirty vineyards. So in terms of strategies, that's a really good strategy. You know, Monica Cooper's working on it, and various other people are, but it really is it's a great strategy if you can do it, to coordinate removal with your neighbors.
Here's a question. What's that? There's so many more. All right. We'll do two. Okay. Could you please comment on the accuracy of Leaf-Roll 3 tests, given that it's highly variant? And some of you mentioned that Leaf-Roll 3 was very hard to detect in rootstock. So in the rootstock, is it just not showing visually, but can you see it in a lab test? Can you talk about the accuracy of Leaf-Roll 3 testing?
On scions, using ELISA. ELISA, the kit we have in South Africa, which we produced, picks up all the variants that are known at this stage. So ELISA is quite reliable, but its sensitivity is not as good as another technique, PCR. PCR is variable in what it can do, depends on how you design the technique. It can pick up individual strains or much wider, but it's more sensitive. So on scion material, I'm not too concerned. Between ELISA and PCR, we've got it pretty much covered. Rootstocks are another ballgame. We're not quite sure what's happening in rootstocks. We initiated the studies on rootstocks because when we were testing them with ELISA, we'd be getting very low absorbance values. When we tested them with PCRs, it would be variable. So that's where we started doing the experiments, where we went to a vine that we knew was absolutely, definitely leaf roll infected, but actually tested the rootstocks there. And in about 2/3 of the instances, we couldn't find type 3 down there, not even with PCR. And we're actually wondering if what we call, say, Richter 99, if that's actually a homogeneous cultivar, and whether there aren't possibly genetic variations in there, some of which are susceptible and some of which are pretty well immune.
Thank you. Last question, although there are so many good ones here. How long does it take for the virus to move through a grapevine from the time of infection? Is cane pruning a better method than cordon pruning from keeping the virus from spreading throughout the vine? Any comments there?
Thank you, my friends up here. I knew they were my friends, right? Actually, there is no easy answer to this one. It's an excellent question, and I don't think there has been, at least I'm not aware of any scientific sound experiment that has been designed and actually implemented to address this question. intuitively, as a virologist with my little experience, I would say it doesn't make any bit of a difference. Regardless of how you prune it, the virus will find its way throughout the whole vine. It might take more time. It will depend on, as I just mentioned, on the particular clone of a sign of your choice onto a rootstock in a given environment. But I don't think that the end effect will differ how the vine is pruned and maintained.
Great, thank you. Stephanie?
Yeah, I would agree with that. Oh, sorry, Stephanie. We farm a lot of cane pruned. Almost 90% of our vineyards in the North Coast are cane pruned, and they're just as susceptible. It moves just as fast through those as it does in cordon prune or machine prune vineyards.
All right, so I know that there are more questions, but we do have plenty of time. All of these guys will be around until about 1:15, so you can interact with them. They're all agreeing to stay through there. Dr. Maher Al-Rawana wanted to mention something from FPS really quickly to the audience too, so if you'll run up here, Maher, but let's give a round of applause for our great panel.
Good afternoon, everybody. I'm Maher Al-Rawana. I'm the lab director at FPS, and I would like just to clarify a couple of points that we learned a lot, and I will second what Mark said and everybody was saying, that it's all our job to work together to ensure that you get the best and the cleanest plant material. Dr. Stamp presented the table out of the FPS newsletter where it shows the testing, and just want to clarify that FPS is not the source of an infected plant material. What we did, like if we plant material out in nature, plant material is get infected. What FPS is doing is to ensure that we test each and every plant before we ship it. And if you noticed, just please visit the FPS website to learn more about what we are doing to ensure that we give you the best and the cleanest plant material with testing and testing and testing. And 2018, 2017, FPS adopted a test to order policy because we have zero tolerance for any virus infecting. And the recent incident of Russell Ranch infection of red blotch, we had history of testing between 2013 when we established the vineyard until 2017 when we start seeing some infection, which we believe it comes with a vector. So I just want to clarify this. Still, we have the source of the cleanest plant material, and each and every vine in both foundation vineyards, Russell Ranch and the Classic Foundation, is tested annually now for leaf roll 3 and red blotch. Thanks for the opportunity for responding.
Yeah, thank you. So I think you can tell that everyone in this room is pretty passionately involved in doing their part to give us, to reduce vectors of viruses and to reduce virus inoculum. And if you want to meet the director of Foundation Plant Services, Deborah Galino, is with us today. She actually rearranged her schedule. She has some visitors, and she rearranged things to be here with us today. And so did several other people, and I'm sure you did too. So take-home messages. We have to take some action. We have to do it together. I think we all hit home the message that it's important to rogue. We'll be learning about that more. We'll hopefully create some more handouts and outreach materials on that. Now it's time to interact with vendors, go to lunch, and if you're invited to one of the afternoon sessions, come see me. I'll give you more information. Thank you all very much. Let's do this.
One of the most impressive things from this morning was to see people brought in from all over the world to tackle this problem collectively.